Hello, Pasangla. Yes, yes. Am I visible? Hello. Welcome all of you to We Need Tamil Nadu. எப்படி இருக்கீங்க? நல்லா இருக்கீங்களா? So, yes, I didn't know I was live. Okay. Suddenly அது click ஆச்சு. So, அதனால it started. Let me just check myself in the phone. Okay. If I'm live or not. Yes. Hello. Hello. Okay. Yes, one more teacher was taking class students, so அதனால தான் கொஞ்சம் delay ஆச்சு and I still can't find the AC remote. So அது மாதிரி கொஞ்சம் problems இருக்கு. Yes, I am live. Okay, let's start. So இன்னைக்கு நம்ம வந்து biotechnology principles அதுக்கப்புறம் application. Okay, இந்த ரெண்டு chaptersமே we are going to decode. So make sure that you are all ready with your pen and your NCERT and you should have focus. Okay, these are the three things that you require for this class, because in the chapters biotechnology principles and application இந்த ரெண்டு chaptersமே high weightage chapters தான். Principles எல்லா years 6 questions come. Okay, every year continuously 6 questions varum. Adukapram நம்ம application chapter nu 4 questions. And are these questions very difficult? Not at all. In the questions வந்து mostly easy level questions ஆ இருக்கும். So அதனால 40 marks வந்து பக்கா. Okay, 40 marks வந்து பக்கா. Clear? Yes, understood? Good evening. I don't know the chapter, ma'am. Please teach. Okay, make sure that you're sharing the video with all your friends. Okay, I know some of them will be still in college and everything. They will take some time to come. But Anna, still I want all of you to share the video with all your friends. Okay, adudha mukkyo. Okay, clear? Start panlama? Very tanam. Okay, awesome. Okay, quickly mark the attendance and make sure that you mark your attendance from everyone's phone. Okay, your mother's phone, father's phone, sister's phone. Okay, neighbor's phone. Run to your neighbor's home also, disturb them also, and make sure that you do all of that. Okay. Okay, very nice. Okay, let's start the class, students. Biotechnology. Okay, so நம்ம இப்போ வந்து இன்ஸ்டாகிராமில் வைரலாக இருக்கிற ஒரு வேர்டு இருக்கு, டெக்னாலஜியா, அதுதான் இங்கே. ஓகே, ஸோ இந்த சாப்டர் வெயிட்டேஜ் பார்க்கும்போது சி ஸ்டூடெண்ட்ஸ் போத் த சாப்டர்ஸ் வந்து ஆட்டோமேட்டிக்காக 40 மார்க்ஸ். இமேஜின் 40 மார்க்ஸ் இன் நீட், getting it directly. Adudha in the class 40 marks, how we are going to try to achieve it. So today concept also will be there, question also will be there. So make sure that you are sitting with a pen and a highlighter also. Okay, pencil and a highlighter. நான் சொல்லது எல்லாமே NCERT side you have to note it down. இல்லைனா மிஸ் பண்ணிப்போங்க students. Later when you open NCERT you will not find anything. But I am not telling you sit and make the entire notes. I will already make the notes and share it with you. Anna, I want you to underline certain things, write it in the side. Okay, clear? Yes. Okay, now let's understand biotechnology. How many of you have heard about this word biotechnology? Biotechnology, ma'am, very simple word, ma'am. I've heard it so many times. When I went to take my PUC admissions also, they told me, do you want biotech? Okay, do you want instead of biology, biotech? Neet students, other than the biology students, biotechnology students also can write Neet. So what is exactly this biotechnology? So biotechnology na students, bio na ena? Ma'am, bio na any living organisms, correct? So bio na living, okay, living organisms. Technology na technique, technology na technique. So nama different techniques use panni, living organism use panni, how can we make it better, right? How can we make it better? So living organisms and technique. So I am using living organism. Okay, I am using living organism, integrating different types of techniques to bring about any changes or to bring about a product. Adudha biotechnology. Ipo biotechnology nama vandu oru funny example la we are going to understand. Okay, I want two people, two names. Okay, not your names, some other names. Okay, I want to make two hens. So give me two names of two hens, whichever names you want, quickly in the chat box. Without wasting 1 minute, I want all of you to answer. So this is what is biotechnology, living organism and technique, and you have a definition to study by the European Federation. Okay, this definition is going to be asked for you, and idu vanda appadiye by heart pannam students. So this also we will be understanding, but let me just give you a basic introduction. Enna ma'am in the biotechnology? Okay, yay, easy peru kudada, easy peru. What you people know, eppadi pandra? Angel, hen ka name Angel. No, Chintu. I'll only time you. Your names are very bad. You people names are like that. Some, I don't know what to tell also. Words are not enough to tell you people. Okay. No need. All of your names are very bad. Okay. All of your names are bad. So we will use the word Sammy. Okay. And Jimmy. Okay. I've used this before also, I guess. Because you people are not capable of giving one name. Okay. Sound is low. Wait. How come? Sound should be high, no? Only for Karishma sound is low? Sound is high only. Okay, more than this sound can't come. Okay, wait, that orange chair is reflecting. Why have a person kept an orange chair here? Okay, tell me this. You people tell me. Okay. 1 second, students. Idella pannano life ille. Okay. Now can you see orange hair? Okay. So look here. Sammy and Jimmy. Okay. So students, now look here. Sammy vanda, it is a very, okay, very hefty hen. Hefty na, okay. This might not look like a hen, but doesn't matter. Okay. This hen, students, it's very like chubby hen, like nama KFC tinna madri hen. Okay. Ana nama Jimmy vanda, okay, nama Jimmy vanda rumba tired, okay, thinna irukira oru hen. இப்போ இதுல என்ன ட்விஸ்ட்னா, நம்ம இந்த hen வந்து it lays only 2 eggs. ஆனா நம்ம ஜிம்மி வந்து it lays in a day 10 eggs. Okay, எவ்வளோ? Okay, whatever, how many ever eggs I have dropped. So then imagine, okay, imagine that Rajkumar is the owner. Rajkumar is like, ade pidida, this one is hefty, this one is so thin, but it is laying so many eggs. So this fellow has some gene. This fellow has some gene that is helping it to be egg laying. This fellow has some gene that is helping it to be muscular. So what if I mixed both of it? So I can get the eggs also. Adukapra in the hen vandu KFC ku anapra. So I am making money. Okay, commercially I am making money. So here what did he do, students? He took, let's say this is the host. Okay, nama Sammy the host. So we have taken the host DNA. Okay, host DNA. Adukapra namma inda Jimmy, we have taken Jimmy's DNA. Okay, just imagine Jimmy's DNA, this much is there. We don't need the whole DNA, we just need one portion which is allowing this Jimmy to lay this many eggs. So this is my gene of interest. Can you see this red color? Okay, this is my gene of interest. So on the gene of interest, I am going to extract. Okay, I am going to extract. How am I going to extract it? With the help of molecular scissors. So I am going to use molecular. Okay, what scissor is this, students? Okay, I am going to use in the molecular scissors use panni. Idu cut pannita. Okay, I am going to take this gene of interest. Now what is going to happen? Raj is making completely a new different species which is hefty. At the same time it can lay 6 to 7 eggs. So enna nadakudhu, students? So this is happening. Now you tell me, in this host, okay, I am going to use a vector. Okay, because host will not directly accept, telling that, oh, Raj wants to make money out of it, so I will directly enter. Adu vandu nadakadu. So adanaala நம்ம வந்து ஒரு vector use பண்ணி, okay, ஒரு plasmid, ஒரு bacteriophage, so அது மாதிரி ஒரு vector use பண்ணி இந்த gene of interest வந்து இங்க insert பண்ணோம். So insert பண்றதுக்கு space வேணுமே? Yes ma'am, space வேணும் ma'am. Space இல்லாம எப்படி ma'am? So அதனால இதுமே cut பண்ணி ஒரு space I am trying to create. Okay, I am cutting this and trying to make a space in the plasmid. Now the space is made. Yes ma'am, space is made. Now what I am going to do, students, I am going to add my gene of interest. Understood? Understood? All of you mark your attendance quickly. Okay, I am going to add my gene of interest. Now I got a new one, right? I got a recombinant DNA, right? I got a recombinant DNA na enna? Recombinant means something new. Recombinant DNA. Now if I will insert this into the host, okay, I will insert it into the host. Now I am going to get a hen which is hefty, at the same time going to lay around 6 to 7 eggs. So is Rajkumar happy? Is Rajkumar happy? Ma'am, Rajkumar is super duper happy. Okay, Rajkumar vandu super duper happy aayirukkranga because the hen is hefty, at the same time it is also laying so many eggs. So this is what is recombinant DNA technology. That means I am using the gene of interest, using a vector and inserting into the host, so I can get a product. I can get a product at a larger scale. How many of you understood? If all of you understood, quickly put thumbs up in the chat box or fire emojis in the chat box. Then we can directly get into what is biotechnology. Did you all understand? Yes or no? Yes or no? Yes or no? Understood, ma'am. Okay, understood? Yes, perfect. Perfect. Happy. Okay, yes. So, students, do you think biotechnology came now? No, ma'am, what are you saying? Some of you will tell, yes, ma'am, biotechnology came now. I have not heard my grandmother or grandmom talking about it. But, Anna, nama biotechnology la vanda, our grandparents have been already using it. Okay, our grandparents have been already using it. Okay, already using it. That is, there is traditional method. Okay, there is traditional method. Traditional method, and there is modern biotechnology. Okay, so in the chapter, whatever you are studying, you are going to use it where? In application chapter. Okay, and I will do it faster, don't worry. So, traditional biotechnology and modern biotechnology. Traditional biotechnology ne enna? Nama parents vanda, have you seen, they take little bit of curd, they boil the milk, and the curd vanda, they will add it in the milk and mix pandranga. Automatically next day vanda adu complete and the milk vanda curd agum. They do not know that there is lactobacillus inside this curd that they added, but the curd that they added as an inoculum converted the entire milk into curd. So traditional method la biotechnology always existed, but ana ivlo importance or terminologies vanda it was not present. So adanal students, traditional biotechnology la we study about how we make bread. Okay, how we make dosa and idli. How many of you like dosa and idli? Tell me in the chats. Dosa and idli la eppa ma'am? Nama yeast add பண்ணும்போது students, yeast add பண்ணும்போது have you seen that fluffiness will come? Bread la yeast add பண்ணும்போது அந்த fluffiness வரும். நம்ம cake எல்லாமே இப்படி ஆகும், இல்லனா cake வந்து flat ஆயிருக்கும். இப்படி it will what you tell, it will swell up. Okay, so that is because again why? Fermentation is happening, carbon dioxide is released, those holes are coming in the cheese, in the bread, in the dosa, in the idli. Adellame our grandparents have found it, but Anna they didn't give the term biotechnology to it. So traditional method le nama bread, dosa, idli, okay, wine, okay, curd, all this is our traditional biotechnology. Anna namma vandu improve aachu. Yes ma'am, obviously we improved it, right? So modern biotechnology la we study something called as genetic engineering, where I am manipulating the genes. Okay, I'm manipulating the genes and trying to make something, like example humulin. Okay, humulin. Understood? This is the introduction. Idhukapram we have to study definition. Okay, very good energy, very good energy. I can only see Deepak and Karishma having energy. Other people are sleeping, huh? Rajkumar also little energy is there. Huh? Other people are sleeping, huh? Class just started. If I have to, get through this, you should have full energy. Okay, yeah. Insulin you have to tell, instead of that you can tell humulin. Okay, humulin you can tell. No, Gautu, you're not late. Okay, so understood this? Understood this? Now, look here. Principles of biotechnology. So, nama NCERT le the principles they have given. Principles of biotechnology, students, you have two things you have to study. One is what I just now used, that is called as genetic engineering. Okay, genetic engineering. Adukapram bioprocessing engineering. This is bioprocessing engineering, genetic engineering. Bioprocessing engineering illana genetic engineering, students. In the genetic engineering da nama in the motto chapter le genetic engineering பத்தி தான் we are going to talk. That is technique where we do gene manipulation. நம்ம வந்து இப்போ recombinant DNA how we made, okay, we made Jimmy and Sammy together, right? We took the good things of Jimmy, good things of Sammy, we made a combination that was going to be commercially used. So அது மாதிரி தான் நம்ம வந்து genetic engineering we are going to do gene manipulation. Again in future you will study in application chapter gene therapy, gene addition, subtraction and the ella words may RNA interference, adhu lot of terminologies where we are playing with the genes, we are manipulating the genes and getting what we want. So in the genetic engineering it's a technique of altering, okay, altering the genes, okay, altering the genes. Clear? Technique of altering the genes da nama genetic engineering. Okay, basically enna pandranga students, nama vandu DNA illena RNA, nama nucleic acid, from there we take a gene of interest, we insert it, adellame panni we are going to insert it into the host, and host la vandu enna nadakum, phenotypic change will happen. Phenotypic change will happen. Okay va? So technique of altering genes, so we can say in simple words gene manipulation. Again I'll teach you in transgenic animals. Adukapram bioprocessing engineering. Imagine panikonga students, you are entering a lab, okay, you are entering a lab, you are like, okay, I have to do this and that in the lab, and once you enter the lab you do not see any sterile condition, and because there is no sterile condition, what is going to happen? Every other microbes also will start growing, right? Every other microbes also will start growing. Now imagine in my class there is no proper proper mode, okay. Let's say I'm not making sure that only good students are coming, or I'm not making sure that spammers are not coming. Some uncle is sitting and sending me hi on the chat box. How will you be irritated? So adu madri oru filtration, oru sterile condition I have to create for all of you to study in my class. Adu madri da in the bioprocessing engineering la nama enna pandrano, genetic engineering whatever method we are doing, and the method ellame oru sterile condition la nadakanam, illena unwanted microbes also will start growing, okay. Unwanted microbes also, yes, Rosie the cow, unwanted microbes also will start growing. So we have to maintain a sterile condition, or I can say in other words a microbe free environment, okay, micro-free environment for what? For desirable growth, for desirable growth of the microbe. நீங்க வந்து இந்த வீடியோ friends கிட்ட share பண்ணாம you people are sitting, many of you, okay, many of you are sitting in the class without marking the attendance, not sharing it with your friends. Now you will tell, ma'am, I don't have friends, I know this, okay, I've heard it a lot. Come on, let me see your attendance. Oh, very bad attendance. Come on, fast, we have to reach the target, okay. Target is already set, now you have to make the target. That is your job, not my job, right? Your job. So maintain a sterile condition, microbe-free environment, desirable growth of the microbe has to happen. So idha nama basic. Now we will see what was EFB telling, okay. EFB vanda what it was telling. EFB na your European Federation, okay, European Federation of Biotechnology. Biotechnology, students, is a technique, it is the definition, okay, is a technique using living organisms or their enzymes for products and processes useful to humans. Correct or not? I can use E. coli, I can use bacteriophage, I can use cancerous cell, I can do disalarming of them and then use it for my own needs. So adu madri da for my benefit I can use anything, correct dane? So look here, the European Federation of Biotechnology, EFB, defines biotechnology as the integration of who? Natural science and organisms or the cells or the parts therefore, and the molecular analogues. Molecular analogues means what? Whatever is there in the molecular level, right? Molecular analogues for products and services. Many students know when they see this definition, they'll feel like that and the Tare Zameen par illana nama Three Idiots le. Three Idiots le madri ellame it will be floating, no? Adu madri tha. Nothing, students. Integration of enna ma'am bio natural sciences of organisms, cells, parts thereof for what? And not just parts, molecular analogues. Genetics I can use, no? Right? Molecular analogues to form products and processes. All of you repeat after me, come on. All of you repeat this definition. In the definition, romba important. In the definition, they can twist a turn, mixing words, anything they can give. Okay, I don't know. If I was NTA, I would have given this definition as missing words. Okay? They'll be like, ma'am, thank God you're not in NTA. Huh? Thank God you're not NTA, ma'am. Understood? Understood? Yes. So, the European Federation of Biotechnology, the integration of natural sciences, okay, of who? Organisms. Then inside organisms, cells, any parts or molecular analogues for processes and products. Okay, va? Understood? Products and services. So, here, now we are going to understand techniques. Okay, techniques of genetic engineering, students. So, here we have understood about recombinant DNA technology. Recombinant na enna? Something new I am trying to form. Something new I am trying to form. Okay, va? So, inga vanda students see, the former provides opportunity for variation and formulation of unique combination of genetic setup, some of which may be beneficial to the organism as well as to the population. Asexual reproduction preserves the genetic information. Here, see, asexual reproduction, usually what does it form, students? Ma'am, it forms clones, ma'am. Correct, thane? Asexual reproduction la variation kediyadu, clone formation da rombo important ana process. So, asexual reproduction and the genetic information save da irukum. Okay, clear? Last unit, which unit, dear? Organism. I have not done that unit. Okay. See, you people, the VNEET Tamil Nadu students are the only ones who are seeing my entire biology videos. Cell the unit of life, I will upload it by Friday. Okay, because many people are asking. Other chapters also, by December, all the chapters will be completed. I will try to do it faster. So, you are the only ones who are going to see my entire biology, because before I used to do only zoology, right? But right now, I am going to do zoology and botany. So, sexual reproduction permits variation. Sexual reproduction permits, enna students? Variation. Correct thane? Yes, ma'am. Sexual reproduction la namakku gametes irukku, and the gametes nama permits, it permits variation. Okay. Traditional hybridization procedure used in plants and animal breeding very often lead to inclusion and multiplication of undesirable genes with desired genes. So, nama traditional method la nama hybrid formation illana nama artificial hybridization pannumbodhu, there are chances that undesired genes also will be growing. You think when you are studying only desired part of your brain is working, undesired brain cells also will be dancing like this. Hey, Instagram check panna, and the crush message panita, okay, and the TikTok video paaka, and the YouTube short poi paaka, okay, Gopika Ma'am video apparama paakalam, okay, adu madri nama unnecessary brain cells also will be dancing in the head. So, adu madri tha, when we do artificial hybridization, there are chances that desired gene will be there. With that, some undesired gene also is there. That is where bioprocessing come into existence. Okay, bioprocessing come into existence. The technique of genetic engineering, which includes creation of recombinant DNA technology, okay, DNA, use of gene cloning and gene transfer. Okay, gene cloning, students, clones. After doing that, okay, we have to form multiple copies. Okay, other than gene cloning. Okay. Overcome this limitation and allow to isolate and introduce only one or set of desired gene without undesirable. So, number one, and the genetic engineering and bioprocessing, whatever we study, same thing they have written here. Okay, clear? No ma'am, it seems. All lie. All lie. Okay? Yes. So, here we are going to start with the first topic. Okay, first topic is discovery of recombinant DNA technology. From that time I am telling recombinant DNA technology. None of you are asking me only where is recombinant. Yes, thanks for reminding. Okay, Pop Scooty. Saturday we are doing a special session where I will be teaching all the NCERT scientists. Okay, all the NCERT scientists, because 2025 question came from scientists and people could not figure out the answer only. So, I will be doing scientists as well as I will try to do summary later. summary vanda namak exam, okay, 1 month before we can do. Anna, this is a very special session. I want all of you to join. Make sure to share it with all your friends. It's important for your boards as well as for your NEET. Very important for NEET. Okay. So here this time we are, you know, shielding ourselves. NTA ask any question, we should be in a position to answer. And the shield da nama fix panna poro. So summary we are going to do, you know, starting page of NCERT. We are also going to do what is there in the cover page of NCERT. Okay, we will see if then one butterfly is there also. We will try to learn that. Okay, so other than our goal. Okay, now look here. Discovery of first recombinant DNA technology was given by Stanley Cohen and Herbert Boyer in 1972. Okay, easy to remember 1972? Yes ma'am, easy to remember 1972. Now what did they do, students? Nama NCERT lines vanda konjo confusing irukku, but ana we will try to understand it. So what they did was, I'm going to teach you, then you will understand. So they took a plasmid. Plasmid na enna? Or extra chromosomal. In cell chapter we study, no? Plasmid is what? Now normally your E. coli is like this. Okay, this is our E. coli, and the E. coli le nucleoid will be there, where the nucleus material is, genetic material is going to be there. With that, there is going to be an extra chromosomal circular DNA. This is our plasmid. So adu madri they have taken some plasmid. Okay, they have taken some plasmid, and on the plasmid, students, there was a region which had, okay, there was a region, I don't think pink will look on green. Okay. Okay, they had a region, okay, they took a region which had antibiotic resistance. Technically, it was tetracycline resistance, okay, but in your NCERT only antibiotic resistance is given. So we will also write that. They took a plasmid that had an antibiotic resistant gene. Adukapram, what did they do? Ma'am, they cut, ma'am, obviously. They cut this, right? Yes, ma'am. Okay, so they cut this using molecular scissors. Okay, yes, they cut this. And now I got the gene of interest. Very important, listen to me carefully, okay? Those who are missing this chapter is going to miss 40 marks, and then you will sit and cry, okay, on May 3rd. Now you are lazy, okay? Now I took this gene of interest. Yes, ma'am, you took a gene of interest which is actually antibiotic resistant. Now students, I am inserting it, okay? I am inserting it into Salmonella typhimurium. This causes gastroenteritis, that is basically your like food poisoning types, okay? So Salmonella it inserts it into Salmonella typhimurium. So what did it do? It took a plasmid which has antibiotic resistant gene, it added it into a host, okay, it added it into a host, and now it is like, okay, this is how recombinant DNA technology happens, right? I took this, I inserted it, okay, I inserted it with another plasmid, okay, now this portion I can take, insert it with another plasmid, yes or no? Yes ma'am, correct. And then I can insert it into a host. That's it. That's all is recombinant DNA technology. Yes or no? This is understood, right? Tell me quickly. Yes, okay. So here they have given the first recombinant DNA was created in 1973 by Stanley Cohen. 1972 is given by Stanley Cohen and Herbert Boyer by cutting the plasmid, okay, PSC101 and a foreign DNA with ECO-R1 and ligating them to form a hybrid plasmid which was then introduced into the E. coli. So what did they do, students? They took the plasmid from the Salmonella typhimurium and then they inserted it again into E. coli. Okay, they inserted it again into E. coli. Understood? From one they took antibiotic resistant, from one they took the rest of it and they inserted into E. coli. This is what happened. I hope this is clear to all of you. Any doubts? Any doubts? Yes, inserted. Okay. So this is the E. coli which is going to act as the host. Understood? No doubts, ma'am. Okay. So we took a plasmid and the plasmid we took an antibiotic resistant gene. Technically it is tetracycline. Okay. அதுக்கப்புறம் we took salmonella typhimurium, we took the plasmid from there. இதில் insert பண்ணிட்டு நம்ம ஒரு recombinant DNA like we have made, அது வந்து நம்ம ஹோஸ்டில் E. coliல enter பண்ணியாச்சு. So இதுதான் நம்ம transformation. So இந்த process இல்ல students, from here to here, this is actually called as transformation, because I am allowing the entry of a genetic material into another cell. So when the cell takes it up, அது வந்து transformation ஆகும். That is why we call it as Griffith's experiment. Remember in Griffith's experiment we spoke about transformation? Yes, that is what is going to happen. And students, in the E. coli one, it will multiply. This E. coli will multiply and I am going to get multiple copies of it. Okay? Any doubts here? Understood, right? So இதுதான் நம்ம first Stanley Cohen and Herbert Boyer வந்து they found it out. First they took a plasmid. So this is basically the plasmid. Okay. அதுக்கப்புறம் they took it from Salmonella typhimurium. Okay. And then they inserted it into E. coli, and now the E. coli will multiply. Whenever something is entering into the cell, that is, we call it as transformation. That means especially your genetic material is entering into the cell. Okay. Now here you should remember, see here, just look here through the NCERT. Let us now focus on the first instance of construction of an artificial recombinant DNA technology. See, if you directly read NCERT students, you will not understand anything. Okay, you will start seeing stars above your head. So அதனால தான் first வந்து நம்ம diagram based we should understand, then we should read NCERT. So look here, the construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistant with a native plasmid that is autonomously replicating circular extrachromosomal DNA of the Salmonella typhimurium, not the typhoid one, Salmonella typhimurium. Okay, so என்ன ஆச்சு, ஒரு plasmid we have taken what? We have taken antibiotic resistance. அதுக்கப்புறம் இன்னொரு plasmidல from Salmonella typhimurium நம்ம அதை insert பண்ணி, அதுக்கப்புறம் நம்ம ஒரு host use பண்ணி, Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistant gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. The cutting of DNA at specific location became possible with the discovery of your so-called restriction enzymes. Okay, that brings us to the next topic, that is your restriction enzymes. I think that went off down. Okay, doesn't matter, we will come to that. Okay, discovery of your 𝐂𝐴R1. Done? Understood this much, right? Now students, let's understand, we are continuously talking about biotechnology, but there should be an order. Okay, or proper order irukkanam, proper tools irukkanam, steps irukkanam. So now we are getting into the actual chapter. Okay, yes, actual chapter. Okay, don't worry, Josh will not go down. I have the Josh. If you people don't have the Josh, it's your loss. I have the Josh. I will teach with full Josh. Lot of students will watch it later. Okay, so look here. First is the basic steps in genetically modified organisms, students. Nama vandu in the steps vannumbodhu there are certain things that we have to keep in mind. So first one vandu identification of the DNA with the desirable genes. Nama Jimmy and Sammy la namakku vandu identify aachu, okay, idukku vandu egg laying capacity irukku, so and the egg laying capacity gene da enakku mukkiyam. So and the desired gene எந்த டிஎன்ஏல இருக்கு அது வந்து நான் ஃபர்ஸ்ட் ஐடென்டிஃபை பண்ணணும். ஐடென்டிஃபை பண்ணாம எல்லாமே கட் பண்ணி சாப் பண்ணி we are not making கிச்சடி or சாம்பார். We need to know properly what we really need. Okay, அதுக்கப்புறம் ஸ்டூடெண்ட்ஸ் introduction of identified DNA into the host. Another introduction of identified DNA into the host. We have to now introduce it into the host. அதுக்கப்புறம் maintenance of introduced DNA in the host and transfer of DNA to its progeny. So this is the basic 3 steps of your biotechnology, that is basic steps. First one you identify pannanum. Okay, let's take an example inga. Okay, first this is the DNA. Okay, in the DNA la இந்த portion da enakku mukkiyam. Okay, in the portion da is important for me na, already you should identify. So first step one enna students, identification. Yes, Joe shouldn't come down. Okay, energy vanda it shouldn't go down, because if you are not typing, if you are not continuously telling yes ma'am I understood, you will actually feel like okay someone is talking and you are in a movie. Okay, you will not take anything to your head. Movie you will take because you paid money for it, Okay, here you will not take. So that's why I'm telling you. Identification done. Adukapra in the desired gene vanda, I have to introduce into the host. How do I introduce it? With the help of a vector. Okay, or a vector use panni, I will add this gene of interest into my host cell. Okay, it's easy to understand. See here, next step on the introduction. Adhikapran students have to maintain this and make sure that the progenies, okay, and the progenies vanda is being formed with the same set of genes. Clear? Like this, okay? I'll have to make multiple progenies. Okay, like this. Understood? All the 3 steps I have drawn and explained you. Okay? All the 3 steps I have drawn and explained it to all of you. Yeah, I know some of you would be in school, but what to do, students? We have to start the class. If I don't start, if I start at 7, the problem is it will become too late. Okay? Yes. Okay, yes. We have just done the basic. Okay, nothing to worry. You have not missed out on a lot. இப்போ வந்து நம்ம ஸ்டெப்ஸ் ஆஃப் பயோடெக்னாலஜி என்னெல்லாம் ஸ்டெப்ஸ் இருக்கு நம்ம பயோடெக்னாலஜியில் எல்லா ஸ்டெப்ஸுமே யூ ஹாவ் டு கரெக்ட்லி நோ பிகாஸ் அன்டில் obtaining of the foreign gene is important for you to know, and these 8 steps are the defining steps of your biotechnology. First step என்ன students? Isolation of the genetic material. That means if I have to take the genetic material, நான் வந்து அதை isolate பண்ணணும், அந்த cell remove பண்ணணும், the outer layers I have to remove. Capsule irukkalam, cell wall irukkalam, cell membrane irukkalam, adu ella layers remove panni I have to get into the nucleus, then I have to remove the genetic material. So isolation is a very important step. Yes ma'am, isolation is a very important step. Adukapra students enna cutting of DNA at a specific location, because if I am telling you that my desired gene is only in this region, I can only cut in that region. I cannot cut anywhere and everywhere. I told you, no, நம்ம வந்து சாம்பார், we are not making sambar, we are basically going to cut the gene of interest. So அதனால if I want to cut it at a specific location, I should remember that I will get fragments. Yes ma'am, you are going to get small fragments, and that fragments only you can use. Correct? No? Correct? No? So then next one is students, separation with gel electrophoresis. So whatever fragments I have got, I am going to separate it with the basis of size. Okay, because there will be lot of it which is not undigested. Imagine, I told you people, okay, I told you people, go there, make sure that you cut all the evidences, okay, that We Need Tamil Nadu is going to become a super hit channel. I told you, I sent you with scissors, okay, you went, some of you looked at each other and like, hey bro, how are you bro, did you eat food and all, which college are you studying, you sat and spoke without cutting any evidence, you came back. Now how do I know if you cut or not? I want to know if that small fragments are digested or undigested. Adu vandu check pandradhukku I have gel electrophoresis. I'll make you people run. Okay, so the thinner one, that is the digested one, will run farther, and the undigested one near the well it will stay. So the third step is done, separation with the help of gel electrophoresis. Adukapra students, amplification of the gene with PCR. Nama gene I am doing one by one. When will I make a bigger product out of it? I want to do vaccinations, I want to do that, I want to do this. So on the process longer aayirukkum. So I will do amplification with the polymerase chain reaction. Okay, adukapra students, ligation na enna, in the DNA and in the gene of interest nama attach pannanume, vector and the foreign DNA attach pannanume. So for that we will need, after cutting, chopping everything, we have to glue it. For that we have DNA ligase. Okay, adukapra students, introduction of the recombinant DNA into the host cell, and last will be expression of the gene and production of desired product. Illena, in your NCERT they have given obtaining the foreign gene. Obtaining the foreign gene. See, if you know this steps, no, then the entire NCERT will be easy. If you don't know this step, then NCERT will be like one world only. Okay, that's what. Clear? Clear? Okay, so this is the steps. Now let's understand tools. Now I am telling you that okay, steps are there, tools are there, but just imagine like you are making something. So நம்ம வந்து biryani பண்ணனும், we have to take rice, we have to take chicken, we have to take tomato, onion, we have to take dhania, pudina, இதெல்லாமே எடுத்து தான் biryani செய்ய முடியும். If you just tell cooker A make biryani, the cooker will come and hit on your head. So அது மாதிரி தான் இந்த recombinant DNA technology பண்றதுக்கு கொஞ்சம் tools வேணுமே. கட் பண்ணதுக்கு ஒரு டூல், அட்டாச் பண்றதுக்கு இன்னொரு டூல், அதை டிரான்ஸ்போர்ட் பண்றதுக்கு இன்னொரு டூல், அதை அக்செப்ட் பண்றதுக்கு இன்னொரு டூல். ஸோ there are some few tools that you have to study. Ready all of you? Okay? Okay? Ready all of you? Are you all enjoying? Are you all enjoying? We are so close to the target. Good job. Okay, yes. So students, tools of recombinant DNA technology. I can never draw a straight line. This is okay. How many of you are there who even after getting a scale you will draw it ulta? I am like that. Okay, first வந்து நம்ம என்ன கட் பண்ணனும். So adukku I am going to use restriction enzymes. I am going to use what? Restriction enzymes. Okay va? To cut, I am using restriction enzymes. Now, to form anything new, I will need what? Polymerase enzyme. You have studied this in molecular basis. Students, you need to study molecular basis of inheritance before watching biotechnology. Illana, you will not understand anything. Because here we will talk about nucleotides, we will talk about phosphate, we will talk about sugar, we will talk about polymerase, DNA, ligase. So, adinala and the chapter vandu clear decode pannum. Okay va? Okay, it is already on the channel, so adinala you can check it. It is in recorded sessions. Okay, adukapram DNA ligase idellame join pannum. Then students, we have to transport. Adinala we need a vehicle da nama vector. Okay, adukapram we need someone who is going to take all this. So adha nama host. So these are the tools that I am going to use. Okay, these are the tools that I am going to use. Okay, here if you want you can write cloning vector also. Instead of just vector, if you feel like, oh ma'am, vector is so boring, ma'am. Cloning vector. Okay. So these are the tools that I am going to use to do my basic recombinant DNA technology. Done, done, done. No, these lectures are not enough for Neet. Then why am I doing lectures for Neet? Da? Every day one question will be there. Okay. Everyone is working in this side means this one person will be the ulta. Is this lecture enough for Neet? Neet 2026 in the editor, we will be teaching what? First standard LKG, UKG lectures. Every lecture where Neet is written, it is enough for Neet. Okay, we are basically teaching you every line of NCERT. After that, you have to solve questions. That is your work. Clear? Okay. Every day one student will be there. Every day one student. One day they'll ask me, ma'am, you will teach for Neet, ma'am. Okay, yes, done. Tools of recombinant DNA technology. Okay, so this is what is given in your NCERT. I'm showing NCERT also, so you people don't cry. Okay, now look here. Now we know from the foregoing discussions, okay, the discussions that we both are having, only I am having discussion, you are sitting there simply. Okay, foregoing discussion that genetic engineering or recombinant DNA technology can be accomplished only if we have key tools like what? Restriction enzymes, polymerase enzyme, ligase, okay, vectors and the host organism. Clear? Full Rajini style, pen and all is flying. Okay, understood this? Okay, these are the 5 things that is required for us to do DNA technology. Now students, we are going to the first step. நம்ம ஸ்டெப்ஸில் பயோடெக்னாலஜி ஸ்டெப்ஸில் வந்து என்ன இருந்துச்சு? ஃபர்ஸ்ட் ஐசோலேஷன் ஆஃப் ஜெனட்டிக் மெட்டீரியல். ஆனால் ஃபர்ஸ்ட் நான் வந்து அந்த டிஎன்ஏ ஐசோலேட் பண்ணணும். அதை ஐசோலேட் பண்ணுறதுக்கு சும்மா if I go like this and tell DNA please give little gene of interest, it is not going to give. I have to cross the paths. Okay, I have to cross many paths. So on the path that we are going to study. Okay, clear? Now, yes. So look here. Treat the bacterial cells. Okay. Ma'am, you are the only one who made the tension into smile. Okay. Thank you. Treat the bacterial cells, plants or animal tissues with enzymes. Like if I am treating a bacteria, okay, I will use a lysozyme. Okay. if I am treating plants, because it has cellulase, I will be using, sorry, cellulose, I'll be using cellulase. So whenever that ASE comes, no, always remember, whenever ASE comes, protease, DNAs, RNAs, nucleases, and the A's meaning enna, and the degrade பண்றாங்க. Okay, they are degrading it. Clear? So chitinase, chitinase, that is fungus. Okay, because fungus, the cell wall is chitin, we will use chitinase to remove it. Now the cell is broken, releasing the DNA and other macromolecules, like what and all, RNA will be there, protein will be there, polysaccharides will be there, lipids will be there. So அதுக்கப்புறம் நம்ம வந்து RNAs use பண்ணி, protease use பண்ணி, lipases use பண்ணி, இதெல்லாமே degrade பண்ணி, only give us DNA, we will tell, only DNA will come to our hand. Okay va? So RNA is removed by treating it with ribonucleases. RNA வந்து with ribonucleases. In the notes வந்து description box ல இருக்கும். Okay, in the notes வந்து description box ல இருக்கும். Proteins are removed by the treatment with proteases. Okay, proteins we will remove with what, students? Proteases. Other molecules are removed with appropriate treatment. So for everything, see, we are, first of all, we are not going to use everything. Correct தானே? We will use only which is necessary for us. நம்ம வந்து எல்லா organisms extract பண்ணோம்னா it is not possible. So அதனால we will have selected. Mostly we will be using our bacteria. Okay, so whatever specialities they have, they will be using from it. So அதனால ஒரு set of enzymes we already have. So from that set of enzymes only we will be using it, students. When chilled ethanol is added, purified DNA precipitate out, collecting fine threads. So now imagine any of you have strawberry in your home. Okay, just take strawberry and you grind the strawberry. Okay, you grind the strawberry. Then inside that you can add water with soap, okay, and salt, okay, and mix it nicely, keep it for some time. After that, into that if you add chilled ethanol, immediately நம்ம வந்து spooling we call it, students. What do you call it? Spooling. So இதுதான் நம்ம test tube. Okay, இதுதான் நம்ம test tube. Okay, so starting la வந்து இது மாதிரி இருக்கும். Okay, with your soap water and strawberry puree and everything. The moment I add chilled ethanol to it, okay, I add chilled ethanol. Okay, when I add chilled ethanol to it, immediately I will start seeing. Okay, I'm drawing it in pink color because இது வந்து already white ஆயிருக்கு. So you will start seeing like this condensed, okay, genetic material. அதுதான் நம்ம DNA. அதுதான் நம்ம DNA. Clear? Who is that? Yeah, I can't read your name. Where is this topic in NCERT? NCERT la வந்து இந்த topic வந்து last page ல. Okay, last second page ல இந்த topic இருக்கு, before your bioprocessing and bioreactors. நம்ம NCERT order வந்து complete ஆன different order, but ஆனா நம்ம order வந்து correct ஆன orderல we will go. Why? Because if you don't understand isolation, then you will not understand cutting, you will not understand separation. So அதனால we will follow a different order. After this, if you read NCERT, you will be able to understand everything. Okay, so students, look here. What is happening? Isolation of DNA is happening. Isolation of DNA la mainly நம்ம என்ன பண்றோம்? First, if you are using a bacteria, lysozyme. If you are using a fungus, chitinase. If you are using a plant, cellulase. இது எல்லாமே யூஸ் பண்ணி நம்ம வந்து அந்த nuclear membrane we are basically removing. Okay, prokaryotes we don't have to do that, but on the membrane we are removing to get the DNA material. So after we take the DNA material, DNA will not come alone. DNA will be like RNA, you also come, protein, you also come. So அதனால அந்த materials remove பண்றதுக்கு protease இருக்கு, RNAs இருக்கு, lipases இருக்கு, இது எல்லாமே யூஸ் பண்ணி completely we will purify it and we'll take only the DNA. And if I want DNA to precipitate properly, I will be doing a process called as spooling, where I will add chilled ethanol, and after the chilled ethanol, slowly with using a like a Q-tip or like a maybe a glass rod, I'll be able to remove out the precipitate. So this is my genetic material. Spooled out. Okay, this process is called spooling. This is also in the end of your NCERT. You can see one test tube where they have taken and shown it. Okay, where they have taken and shown it. Clear? Who is that one conjuice in the class? One conjuice. Okay. Now second one process enna students, cutting. Okay, நம்ம வந்து isolate பண்ணியாச்சு. இப்போ இந்த DNA ல எங்கே genetic material, gene of interest இருக்கு, அது வந்து நம்ம cut பண்ணணுமே. So for that we need cutting people, right? We need a molecular scissors. who was, or how did we find this molecular scissors? How did we know that restriction enzymes were there? So for that, there is one experiment given in your NCERT which talks about E. coli. Now, E. coli bacteria, when bacteriophage, okay? So listen to me carefully, very important. Okay, first we will, okay, this was our E. coli, okay? In the E. coli, students, one bacteriophage. As usual, he came and attacked. Okay, he came and attacked. So when he attacks, what is the first thing that he will do? He will try to release his genetic material into the E. coli. Correct or not? Yes, ma'am, very correct. Adada nama virus. Okay, so here we have E. coli's genetic material. Here we have the plasmid. Same color we are not supposed to do. Okay, plasmid. So idha nama E. coli. So this bacteriophage attacked this E. coli. It released the genetic material. E. coli was the next level. Oh, target achieve, very good. E. coli is next level. E. coli is like, I know to protect myself. So, in the E. coli one, the students, for its self-defense, self-defense, for its self-defense, it is releasing two enzymes. Okay, listen to me carefully, very important. It is releasing two enzymes. If you are thinking that after this I'll go like, okay, no need to listen to the class, I'll go read NCERT, you will not understand anything in NCERT. Okay, complete Tare Samipar Madri and the chapter vanda ulta pulta it will be going. So that's why listen to me carefully. So students, for self-defense in the E. coli vanda, it did two things. First thing it released was DNA methylase. Okay. So basically, first what did it do, students? Adu vanda its own genetic material protect pandradhukku. Okay, to protect its own genetic material, it started adding methyl group. It started adding methyl group to its own genetic material. So the bacteriophage will not cut the genetic material. Okay, cut the genetic material. Okay, wow. Adukapram, it released something called as nama restriction enzymes. It released something called as nama restriction enzymes, which had the ability, okay, which had the ability to cut the bacteriophage material, cut the genetic, viral genetic material, right? Cut the viral genetic material. Purinjada? Understood? So, for the self-defense, E. coli vanda, it did two things. It released DNA methylase, which was adding methyl group to its own genetic material. So, bacteriophage cannot identify only. If it identifies only, it will cut, right? If it doesn't identify, how will it will cut? Okay. So, adukapram, it also released restriction enzyme, which was able to cut the bacteriophage. Understood all of you? Huh? Understood all of you? Yes or no? Yes, methyl group was added. Yes. So, this is how we found the restriction endonuclease. Okay, we found out what, students? Restriction endonuclease. Is this clear? Clear, clear, clear. Clear, clear, clear. Yes, ma'am, super clear. Okay, we got the restriction endonuclease, sorry. Restriction endonuclease. Okay, wow. So, either the self-defense that led to the finding of our restriction endonucleases. Okay, this is the first thing. This is understood. E. coli has a built-in restriction modification system to protect itself from the bacteriophages. Okay, protect itself from the bacteriophages. Very good, very good. All of you understood. Now we are going to understand the working of restriction enzymes. That is, how is this restriction enzyme going to do? Okay, okay, we'll shift it here. Working of restriction enzyme. First of all, students, you should understand this. In the restriction enzyme, it will first scan the DNA because it has to make cuts. Yes, ma'am, it has to make cut. Before that, you should understand this comes into this restriction endonucleases, exonucleases comes into a bigger group. Okay, bigger group of enzymes which is called as nucleases. Which is called as nucleases. Okay, and the nucleases you will have two of them. Okay, 2 of them. Endonucleases and exonucleases. Endo and exonucleases. Understood? Nucleases na nucleases. Nucleases na I told you when aces come, it is cutting, chopping something, right? Something is happening. So, adanale here you should understand these people are going to break the phosphodiester bond. Yenna phosphodiester bond, it is between who and who? All of you tell me in the chat box. Phosphodiester bond, students, it is between 2 nucleotides. Namma vanda we have understood there, no? That is, one nucleotide is there, another nucleotide irukku. Between these 2 nucleotides, there is phosphodiester bond. And the phosphodiester bond, this is going to cut. So, we have endonucleases and exonucleases. Example edukala, students. Okay. Look here. This is one strand. Okay. This is another strand. Clearly, you should understand everything. I don't want any student to leave this class without having a clarity on both the chapters. Okay. Clear? So, okay. So, this is the 2 strands. Okay, va? This is the 2 strands, students. Now, endonuclease vanda, what is it going to do? In the endonuclease vanda, students, it is going to cut within the DNA. It is going to do what? It is going to make the cut within the DNA. That means in between, okay? In between the DNA, it is going to create a cut. So, endona within, okay? Endona, yenna students? Within the DNA. Within the DNA. Okay, va? Understood this? Exona yenna? Exo, washing the plate, okay? Exona yenna? It is going to cut at the end of the DNA, okay? Only at the end of the DNA, இது வந்து correct cut பண்றாங்க. ஆனா இது வந்து within okay? எனக்கு வந்து ஒரு small portion of gene necessary na இது வந்து cut பண்ணி it will give it to me telling that take bro, you take whichever, okay? நீ எடுத்துக்கோ, okay? அது மாதிரி endonucleases வந்து within cut பண்றாங்க. Exonuclease வந்து only at the ends it is going to cut. Clear? Understood this? Yes. Okay, super fun. It's like our friend we are talking, no? It's like we are talking to one of our friend. Yes or no? Yes, ma'am. Correct. Huh? Correct. Okay. Sure. Okay. Understood this? Yes, very good. Okay. So, endo and exo, okay? Endo and exo. Now, we have to still study about the working, okay? We will come back to the working. Once again, we have to, before that, we have to study one more topic, which is the nomenclature. Here it is. Again, working is here, students. So, yeah. So, working, how does this people work, ma'am? Okay. So, students, first one I told you already, if they have to cut something, right? If they have to cut something, adu cut pannanamna, adu vandu first scan pannanume. Without looking at the paper, you will cut or what? You can't cut. Scan pannam. So, first they will do inspection. They will inspect the strands, okay? So, you can also say scanning, okay? You can also say scanning. So, the first step they will do is inspecting. First thing that they will do is inspecting the strand. They will go through the strand and they will see, okay, where the cut should be made. Okay, where the cut should be made. Check at the working of restriction endonucleases. Stand C. Okay. Yes. So, they will do inspecting the DNA strands. Okay. Then what they will do, students? They will bind to that spot. Okay. They would have found one sequence. Okay. So, they would have found or identified, I'll tell. You don't have to find this in the NCERT. This is, see all the steps I am giving you, so your understanding becomes better. Because NCERT does not care about how much you understand. NCERT cares about giving you the information. Ana, it's my job to make sure that you understand in a flow. So, adanale, in the topics ellame, I'm putting that add-on topic so you understand the entire flow. See, now you're able to relate. Okay, isolation of DNA happened. Now we are going to do cutting. Cutting restriction endonuclease we found from E. coli when it attacked bacteriophage. After that, I understood this restriction enzymes or this endonucleases comes into a bigger category of enzyme called nucleases. Endonucleases, exonuclease. Endo will cut within, exo will cut at the end. Understood? Nucleases. Nucleases means something it is breaking. It is breaking what? The phosphodiester bond between both the nucleotides. So, the flow is very clear. Understood? Now I am telling you how they are going to cut, because I told you one can cut within, one can cut at the ends. But how do they cut? First, they have to scan the DNA strands. Adukapram, they will identify a certain sequence. Every restriction enzymes will have a certain sequence, telling that I will only cut at this place. Otherwise, students, anything can cut at any point, right? So, I will cut only at 1, let's say at 1 o'clock. I will only cut at 2 o'clock. Adu madri or specific sequence irukum. So, they will identify a specific sequence, okay, specific sequence, and then they will bind to it, they will bind to the sequence and then cut at the region, cut at the particular region. These are the 4 steps, okay va? Understood? Okay, clear? So, now I told you they will identify a specific sequence. Yenna ma'am in the specific sequence. So, students, imagine this is a sequence, okay? Now, like this sequence that I am talking about is called as palindromic sequence. Another palindromic sequence. Palindromic sequence na oru 6 base pairs repeating unit da. Ana, you read it from this point to this point. That is, you read it from 5' to 3' or from 3' to 5'. It will be the same. It will be the same. Complementary will be there, otherwise it will be the same. Example like mom. Mom. I read it this way also it's same. I read it this way also it's same. Illena, dad. I read it DAD also correct, DAD also correct. Illena, Malayalam. Okay, my mother tongue. Malayalam. Students, MALAYALAM. MALAYALAM. Right? So many words are there. So many words are there. Like wow. Okay. Okay. So, this is called as palindromic sequences. Palindromic sequences are specific regions where the enzyme is going to cut. Otherwise, everyone can start cutting at every place. No. So, adanale here you see GAATTC. Okay. In the GAATTC, students, what is going to happen? CTTAAG. Okay. CTTAAG. Okay. So, here if I write it G, T for A, T, 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 C. So, if I read it this way also it is same. If I read it this way also it is same. Correct? No? GAATTC. GAATTC. So, from 5' to 3' it is the same. Okay? From 3' to 5' also it is the same. Clear? These are palindromic sequence. Yes, specific recognition site. Very good. Very good. What do you call this? Specific recognition site. Specific recognition site. Understood this? Yes or no? Yes, ma'am. Understood. Specific recognition site. Okay? Yeah. Good job. Okay, example understood. So, in the sequence vanda, it is actually a sequence of your 𝐂𝐴𝐴. 𝐂𝐴𝐴. In the sequence la 𝐂𝐴𝐴 vanda correcta it will cut. So, what did I tell you? they will identify a sequence, and the sequence the specific recognition site, also called as palindromic sequence. In the palindromic sequence, speciality enna 5' to 3' also it will be read the same, 3' to 5' also it will be read the same. 𝐸𝐶 strand வந்து GAATTC, அது opposite எழுதியாலும் it will be GAATTC. okay வா? அதுக்கப்புறம் students நம்ம hind 2 also has a sequence. Can anyone tell me in the chat box? Hind 2 also has a sequence. Can anyone tell me in the chat box? Let's see how many of you know it. Hind 2 also has a sequence. Let's see. GTT, something like that. Is there or it's not there? Okay, I can see the chats here. Yes. Quickly subscribe to the channel and smash the like button, okay? All of your even individual support is very important for us. Okay, very, very important for us. Some conjuice is not marking the attendance. Okay. Hind 2, anyone? Come on, yaar. Oh, how many of you don't know? I'm going to teach anyways, but still I want to know how many of you know. It will be happiness when I see students knowing it. Okay, that is important, no? Yes. Ha, correct. Don't know, ma'am. Okay, no problem. So, students, hind 2 vanda G, T, we can write it Y. Why? Because in the Y vanda it will change. Okay, in the Y vanda it can change. It can be a T or it can be a C. Okay, adukapram AC. So, this Y can change. Okay, that is why it is written like this. It can be written as Y, R, A, C. That is Y can be C or T, R can be A or G. Okay. Y, it can be T or C, okay. R na, I am writing it R here, R na, it can be A or G, okay. Adukapram A and C, okay. This is the sequence, this is the complementary you have to write. So, this is how it will be written, okay. G, T, T, C or A, G and A, C, okay, done. Yes. Okay. So, now students remember this palindromic sequences are usually 6 base pairs. Okay. 6 base pair sequence da. So, on the sequence when the repeated, it will be repeated. Okay. It will be repeated. Now, next one we are going to study rules of nomenclature. How did I say 𝐸𝐶? How did I say hind 2? Right. That is important. No. So, look here. The convention of naming these enzymes is the first letter of their name. So, example one we are going to take hind. Okay, hind 2. Okay, hind 2. Hind illa students in the H one. Okay, H stands for hemophilus. Yo, this is dancing. Hemophilus. Okay, IN stands for influenzae. Okay, and this D stands for, okay, I should write it here only. D stands for strain. Okay, and 2 stands for discovery. Okay, discovery. So, here you can see, adu madri in the NCERT le they have given, okay, NCERT le, it's coming into chemistry, yes, NCERT students here they have given, enna kodutitanga, 𝐸𝐶 they have given, okay, 𝐸𝐶 they have given. Here, 𝐸𝐶 le, your 𝐸𝐶 comes from, okay, 𝐸𝐶 comes from Wuchereria coli, that is your E and CO is coming with that. Then we have RY13, okay, RY13 where R is derived from the name of the strain, okay, R is derived from the name of the strain, that is the rough strain. And students, this number 1, 2 and all, remember it can be order of isolation, okay. Here I have written discovery, you should remember sometimes in some of them it will be order of isolation, like example in 𝐸𝐶, okay, all the letters came back, all the letters didn't come back, I guess. Students, in the board one it is missing some letters, okay, if that letters doesn't come back, let me know. Wuchereria, okay, coli, Okay, R is the strain. This one the rough strain. We can write RY13 strain also. Adukapra in the 1 the order of isolation. Discovery na enna? Now imagine I discovered that here water is there. But Anna, I did not isolate it. So adanale order of discovery, order of isolation. Okay, come on, energy is going low. Come on, increase the energy. All of you in the chat box. I want high energy. Okay, high energy. Quickly. High energy should come. Fast. Yes, very good. Okay. Clear? Yes. So this is the nomenclature. Now students, one more thing that we have to study is that how these restriction enzymes are going to cut. Okay, how these restriction enzymes are going to cut. That is also important for us. So adanale we will duplicate in the slide one, we will duplicate. Okay, and we will erase all of this. Okay, don't worry, it's already saved. Come on. Okay, yes. See students here, how do they cut? Okay, how do the energy is awesome. Okay, how do they cut? So here let's just write it again, GAATTC. Okay, GAATTC. This is our sequence. Okay, idhila ipo cut vandu eppadi nadakkum? How does the restriction enzyme cut? Adu mukkiyam thane, right? What kind of cut it is making? So students, some of the restriction enzymes like your EcoR1, அது வந்து கொஞ்சம் different cut பண்றாங்க. எப்படி ma'am? Like example students, அது வந்து இது மாதிரி, it is going to cut like this. How it's cutting? It's cutting like this. It is leaving one hand here, it is leaving one hand here. You understood? Now if I separate it and show you, how does it going to look like? GCA A. Okay, this is one side. Yes or no? Yes or no? This is one side. Correct? Here one G is left, here one G is taken. Okay, so this is one side. Adukapram இன்னொரு side இருக்கு. That is what? A T C G. So if you look at it students, if you look at it, it looks like two missing blocks. Right? It looks like one block like this and it looks like one block like this. Correct ma'am? Ippadi fix pandradhukku romba easy. Right? So what is the meaning of this? That means this is going to cut. That is your certain restriction enzymes are going to cut and they are going to form sticky ends. Sticky ends. Okay? Or overhanging ends. Friends, even though this lecture is done at 5 hours, don't worry, nothing we are going to skip. Nothing we are going to skip. Okay, we are only going to study more. And cohesive ends. Okay, cohesive ends. Sticky ends na enna? Sticky means ready to mingle types. Okay, ready to mingle na enna? Both the sides are sticky. You just imagine, you have one magnet, you have one more magnet. These two magnets will always want to come together. Yes ma'am, makes sense. Correct. So, adu madri da, this is sticky ends, this is also sticky ends. So, idu vandu attract pandradhukku easy. So, in the EcoR1 la illana nama endonucleases la oru rule enna students, if I am cutting the foreign DNA, if I am cutting the vector also, both should have sticky ends. One cannot have different end, one cannot have different ends. Both should have the same end. Otherwise, that matching will not happen. இப்போ இது பார்க்கும்போது இந்த மாதிரி ஒரு block இருக்கு. So that means what? We got sticky ends, overhanging ends or cohesive ends. இதுல example one என்ன students? The example is going to be here your EcoR1. EcoR1 will cut like this. Okay? It will cut to form sticky ends. Clear to all of you? Yes ma'am. Okay? Clear. Thank you, Now fall. He's going to cry now, telling that this ma'am always calls my name wrong. Okay, now students, this is the second sequence that I have. Okay, now you tell me T will code with A, C will code with G at Gopika Ma'am's class. Okay, this is the sequence. Just check if I'm correct. A, G, A, C, H, C, T. Now, some of the people, okay, they will cut right at the center. They will cut right like this, making them blunt. Making both the ends on the blunt irkum. So, adanale G, T, okay, A, G, A, C, A, okay. This one end. This another end. Okay, va? Understood, Kana? Huh? Understood this? Okay. So, this one the students, this one the blunt ends da. Idhila one the sticky ends kediyadu. This one the join pandra vaipun romba kammi. Okay, because this one the blunt acha. So, this madri or irunduchu. Imagine this one the ipdi split aachana, ipdi attach pandradhukku easy. Illana ipdi irunduchuna, ipdi vadda attach pandradhukku easy. Ana inda madri irundha pattern like sorry, Malayalam came off. Okay, inda madri irunduchu sudden cut paniya ipdi agum. So, this one the blunt ends. Okay, so certain of them, certain restriction enzyme will cut like this also. Okay, certain will cut like this also. Clear? So, what do you call this ends as? We call it as blunt ends. Blunt people means what, students? Okay, I took this example before also, or we call it flush ends. Example, boys kitte, if I ask a question, avaru vandu directly yes or no solranga. Okay, ana girls kitte adhe vibe, if I ask, they will tell, no ma'am, I was thinking like this, then you thought that you and don't know ma'am. Correct dane? All the boys will be like, yes ma'am. Full committee they will make. Okay, full committee they will make. Clear? So, idu madri da. Sticky ends, ready to mingle. Okay, ready to mingle. Boys on the direct, yes sir, no, yo ma'am, no ma'am. Padikada ma'am, no ma'am, yes ma'am. So, blunt. Answer on the blunt. Adukapru or question to ask also, I don't have any question. But girls appadi illa. Ma'am, I went to this, don't know ma'am. So, adu madri da sticky, ready to mingle types. Okay, clear? So, this is the way to remember it. So, some of them will cut at this point where it is ready to mingle. It can attach with a foreign particle or foreign DNA or a vector or a plasmid. And this one the blunt ends. It will be very difficult to make them attach. Okay, clear? You can remember small one here example. SMA1. Many are there example, students. Whatever you want, you can search and add. So, this is namma depending on the restriction enzyme, depending on their cut. Okay, what kind of cut are they going to make? Okay, what kind of cut are they going to make? Oh, dun 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 dun. Okay, I'm deleting the unnecessary slides. Okay, this is our NCERT. Quick NCERT we will read. So, in the year 1963, the two enzymes responsible for the restricting the growth of bacteriophage. Adanalada the name restriction endonuclease, because it did not allow the growth of bacteriophage. It restricted the growth of bacteriophage. Adanalada the name vanda. Okay. In E. coli were isolated. One of these added the methyl groups to the DNA, methylase, while the other cut the DNA. Okay, that is how we got the restriction endonuclease discovery with the help of E. coli self-defense mechanism. The first restriction endonuclease was HIND2, whose functioning depended on a specific DNA nucleotide sequence which was isolated and characterized 5 years later. It was found that HIND2 always cuts the DNA at a particular point. Okay, by recognizing a specific sequence of 6 base pair. So, in the number one, you have to remember, students, otherwise, on the 6 base pair, you will write 16 base pair and you will come home. This specific base sequence is known as recognition site for HIND2. Besides HIND2, today we know more than, in the numbers important, the 900 restriction enzymes that have been isolated from 230 strains. So, 230 strains in a namakku vanda 900 restriction enzymes on the isolate ஆச்சு. Okay, of bacteria, each of which recognize different recognition sequence. Is this numbers clear to all of you? Okay, now Roman numbers following the names indicate the order in which the enzyme was isolated from the strain of bacteria. So some places you will see order of discovery, some places la vanda order of isolation. Isolation na na already adu extract panni எடுத்து வச்சேன், பட் ஆனால் ஆர்டர் ஆஃப் டிஸ்கவரினா ஓ ஐ நோ தட் திஸ் இஸ் தேர், பட் ஆனால் ஐசோலேட் பண்ண மாட்டேன். ஸோ அந்த மாதிரி. ஓகே, ஸோ restriction enzymes belong to a larger class of enzyme called as nucleases. Two types of nuclease is the exonuclease and endonuclease. Exo removes nucleotides from the ends of the DNA. Endonucleases will cut at a specific position. Each restriction endonuclease functions by inspecting. Stanzi was asking, no ma'am, where ma'am in NCERT? See, it's here in NCERT. Stanzi understood? So we read every line of NCERT, every dot of NCERT, then we make notes. So first we read NCERT, adukapram we reread NCERT, adukapram we start writing. Okay, even I make a note, students, not just you, even I make notes. Every time I study also I go through my notes. Okay, so inspecting the length of the DNA sequence, once it finds out a specific recognition sequence, okay, it will bind the DNA, cut each strands of the double helical at a specific point in their sugar phosphate backbones. Okay, each restriction endonuclease recognizes a specific palindromic sequence. Understood this? Yes or no? Okay, done. See here, this is how it is. Okay, look here, vector DNA, foreign DNA. I told you, if we want to make Jimmy and Sammy's DNA combined, Jimmy cut also should be same, Sammy cut also should be same. One cannot be sticky end and one cannot be blunt end. So adunala நான் வந்து சேம் ரெஸ்ட்ரிக்ஷன் என்சைம் சேம் மாதிரி கட்டு தான் பண்ணுவேன், இல்லைன்னா அந்த ஃபியூசிங் நடக்காது ஸ்டூடெண்ட்ஸ். இது மாதிரி இருந்தால் தான் ஃபியூசிங் நடக்கும், இது மாதிரி இருந்தால் ஃபியூசிங் நடக்காது. ஓகே, ஸோ வெக்டர் டிஎன்ஏ அண்ட் ஃபாரின் டிஎன்ஏ. ஸோ when I'm cutting, I'm using eco R1 and I'm cutting in such a way that I'm getting sticky ends and here also I'm getting sticky ends. அதுக்கப்புறம் இந்த both the sticky ends வந்து like when I attach it with the help of DNA ligase, I'm going to get recombinant DNA technology. Okay, understood? Palindromic sequence you understood, no? Malayalam is the example. You read it from 5' to 3' or 3' to 5'. அது வந்து same தான் இருக்கும். Okay, both the sides the sequence is going to read the same. Clear? One second. Here also same thing what I've told you. See, 5' to 3' and 3' to 5', you have GATTC, CTT, AAG. Restriction enzymes cut the strand of DNA a little away from the center of the palindromic sequence, but between the same two bases on the opposite strand, this leaves a single stranded portion at the ends, right? Here one G was left, here also one was left. So adu madri. Okay, then okay, yeah. Now this leaves single stranded portion at the ends. There are overhanging stretches called as sticky ends. Okay, I'm just showing you where it is in NCERT, so you don't have to search. Okay, which are named because they form hydrogen bonds with their complementary cut counterparts. This stickiness at the ends facilitate. So they are made the hydrogen bonds. So anytime they are ready to attach. So adinala da in the sticky ends ko speciality because they form the hydrogen bonds. Okay. Now restriction endonucleases are used in the genetic engineering to form recombinant molecules which are composed of DNA. So whatever I taught you, I went through quickly through NCERT as well. So you don't have any doubt. Here also see, restriction endonucleases are used in genetic engineering to form the recombinant molecule which are composed of the DNA from different sources. When cut by the same restriction enzyme, okay, the resultant DNA fragments will have the same kind of sticky ends and these can be joined together end to end by using DNA ligase. Clear? Understood? Whatever I taught you, I just made you read NCERT now. Okay. Now next tool that we are going to take is, students, DNA ligase. Okay, we'll take a water break. 1 second. One second. Easy, no? No one should say it's difficult. Okay, no one should say it's difficult. Yes. Okay, next one is going to be, students, நம்ம DNA ligase. DNA ligase na enna? DNA ligase na நம்ம glue, ma'am. Okay, DNA ligase. DNA ligase na நம்ம glue. Okay, so if I have cut something, let's imagine. Okay, okay, இதுதான் நம்ம DNA, இதுதான் நம்ம gene of interest. Okay, now I have cut it. Okay, now I am waiting to add it in this plasmid. Okay, so I have my gene of interest, இது வந்து நம்ம plasmid. Okay, so now students, the next step is to make sure that I combine these two pieces. That is, I have here one gene of interest, yes or no, and here we will have a plasmid. That also I would have cut with the help of restriction enzymes, and now the job is to make them come together. Okay, அதுதான் முக்கியம். So அதுக்கு தான் I am going to use நம்ம DNA ligase. Okay, நம்ம என்ன யூஸ் பண்றது? DNA ligase. So DNA ligase is a gluing enzyme. DNA ligase வந்து என்ன இருக்கு, students? It's a gluing enzyme. So in the glue use பண்ணி I am going to attach this gene of interest. Okay, this gene of interest into it. So DNA ligase will do the ligation part. என்ன பண்றாங்க? Ligation parts. Okay, so it will do the ligation part. It will form the phosphodiester bonds and it will help in the attachments. Okay, it will help in the attachments. Bonds. Clear? This is super easy, no? Ligation one everyone knows, ma'am. Ligation is so easy. Okay, it's just gluing. It's just a matter of gluing. Okay, many examples are there. You can remember T4 DNA ligase. Before also I have told you this example. Example T4 DNA ligase. Okay, T4 DNA ligase. Okay, va? Next, we'll go to the next one. Okay, next tool is going to be, students, your DNA polymerase. Okay, there's nothing much to study here because DNA polymerase you have already studied in your molecular basis of inheritance. DNA polymerase na the one that is going to help in the enna nadakkudu, full dance it is doing, DNA. Look at the board is like I don't want to study. Okay, so students, this DNA polymerase is going to synthesize. Okay, it is going to synthesize number new strands. Okay, synthesize the new strands. See, the same thing we have studied in molecular basis of inheritance. Yes or no? Yes, ma'am. There also it was synthesizing new DNA strand, but that fellow had too much of, attitude there. Okay, too much of attitude he had. He was telling that I'll only work on a single strand, I'll do polymerization only from 5' to 3'. அது மாதிரி ரொம்ப demand இருந்துச்சு, right? So here also he is going to have demand, students. It's not like he's not going to have demand. Okay, he's the actor at the end of the day. Okay, actor கிட்ட, okay, what are you saying? Actor வந்து எப்பவுமே actor தான். Okay, so imagine, students, one small portion we have to add. Okay, okay, we have to add some strands. Okay, just imagine here. Okay, here. Now if we have to form that wherever you can see the gap, no, wherever that gap is there, your DNA will be like, don't worry, I will do. Okay, it will come and it will add the entire sequence. என்ன நடக்குதுன்னு தெரியாது, என்னமோ நடக்குது இந்த போர்டுல. Okay, like this the new strands will be, the new nucleotides will be added, the nitrogenous bases will be added, or your bases will be added. So this job will be taken care by your DNA polymerase. Clear? Understood this? Yes or no? Like one Indian flag I have drawn. Yeah, so they will form new DNA strands depending on the complementarity. That word doesn't come, students. Okay, so complementary sequence. So if here G is there, it will add here C. If here A is there, it will add G. How will you remember? Add to Gopika Ma'am's class. Okay, remember that trick forever. Clear? This is your DNA polymerase. Next one, students, we are going to understand the vector. Vector vandu romba important. So adanala I want all of your undivided attention. If you want, I can give you a 5 minutes break. Okay, I can give you a 5 minutes break. So all of you can just refresh, and I'll go fill my water bottle and come. Okay, just 5 minutes. Just 5 minutes. Board doesn't want to give you people break also. It's like 5 minutes is too less. Give me more break. Okay, just 5 minutes. I'll be back by 6:55. Okay. Oh wow, that's an awesome target, Deepak. Next target podungo chat box illa. Next target.
See my camera? See, here is a display. Press that button in your camera. Then, yeah, it's okay. But I should see the display now. Like, see, you can see here, I should see this ISO, picture, everything I should see. Then only I can adjust it. For that reason, I'm telling to press display. Like you should see and you should do it, ma'am. Go backside it. Yeah, take a chair, stand it on the chair. Oh, then how can I fix it, ma'am? Okay, what you will do now? See, at least your front side. See, can you see this? Bro, here. Yeah, just change it that at least. Yeah, change it. Show me the display and you change it. Show me this display and you change it. Yeah, display visible, you change it. Like it is, okay, 80. Make it 50. Make it 50. Change it. Now it is 100. Don't make it 100. Reduce it. Reduce. Still you reduce it. Make it 50. Yeah. Oh, okay, fine. Now, see, in front there is another option. Shutter. Yeah. Show me the display. You change it. Like I'm unable to see the display. Yeah, like it is blur to 8.0. Change it, change it. How much it is showing now? 50 is different. That is shutter. I'm talking about that 7.1. Make it 5.6. Reduce it. Yeah. Wait, 1 minute. Like it is. See now. Show me the OBS. Too bright. Okay. Make it the shutter speed. This one as a 90. This one. This is the front one. And the back one is exactly 90. Yeah. You're increase it. Make it 90 or 100. Yeah. Now see. Yeah.
Hi guys. in middle one sir came and he was adjusting the studio. So that is what happened. Okay. I had gone just that side and then this happened. Okay, let's continue. Okay. Yes, hi. We are back. Okay, I filled water and came. So now we can complete this chapter. Okay, start Pannama. Let me see if all of you are back. Okay, yes, some of the students are still coming. Hello, ma'am. Hello. Okay, yes. Okay, next topic on the students, very important on a topic, vector. Okay, vector na enna vehicle. Vehicle na enna nama car madri, two-wheeler madri, cycle madri. Adu vanda to enter into the host, because host vanda, the moment it gets to know that a foreign particle is entering, it will start acting. So we need someone who is going to allow the entry, who is linked with the host, who has a high copy number, who can give me more gene products. So adu madri I need one person. That is going to be my vector. Understood? That is going to be my vector. So vector is basically a vehicle. Enna oru vehicle da. It is the one who is a carrier vehicle. Okay, carrier vehicle. It is going to carry. What is it going to carry? Adu vanda it is going to carry the foreign gene into the host. Okay, foreign gene into the host. Okay, va? Foreign gene into the host. Be it anything. My foreign gene means what? It can be my gene of interest, right? The one that I took from Jimmy. It can be my gene of interest. Correct da ma'am. Okay, nama vector vanda. If students, you tell me, if you want to be a NEET aspirant, if you want to be an AIMS student, can you be like every other student? Can you just study 1 hour, 2 hours and then be like, okay, I'll go write NEET, I'll get 250, 300 marks, I'll be fine with it? Mudiyada. If you are aiming for AIMS, namakku vanda certain categories irukum, certain criteria irukum. So, அந்த எல்லா criteria மே tick பண்ணி தான் you can enter into AIMS. அது மாதிரி தான் ஒரு vector, I am giving such a precious job to this vector, telling that you carry my foreign DNA, you take and go and you give it to the host. So, I need that vector to be a good vector, right? So, a good vector or an ideal vector, ideal illena or good vector vanda, it should have few characteristics, like few criteria. Okay, few criteria. Yenna ma'am, first criteria vanda students, high copy number. High copy number. Yenna idu high copy number na enna? High copy number na or simple example. Like some of us will have notebooks, no? Some of us will have 2 notebooks. Some of us will have 20 to 50 notebooks. Adu madri the high copy numbers. How much the content is there? Genome content is there. Adu the copy number. So there will be plasmids who have 20 to 50. There will be plasmids who have 1 to 2. Adu the copy number. Okay, if the copy number is high, then the transport to the progeny is much easier for me. Okay, so high copy number. Adukapram students, it should have a very good linkage. Okay, linkage with the host. Okay, linkage with the host. Adukapram, it should, see, high copy number irukku, linkage with the host is there. Other than that students, if it has certain selectable markers, okay, if it has certain selectable markers which is going to help me, okay, selectable markers which is going to help me for what? To separate recombinant from non-recombinant. Recombinant from non-recombinant. So, selectable markers is that it will be a hit plasmid. Okay, and students, they should have ORI site. ORI site irukku. Okay, and if they have more cloning sites, all those are an add-on, but Anna, this 3 is very important. High copy number, they should have linkage with the host. If they have the ability to easily help us differentiate recombinant with non-recombinant, then I am going to accept this vector. Okay, are you going to accept this vector? Okay, yes or no? Come on. We have reached till vector. I want all of you to be very, very fast. Okay, you can call this vector as cloning vector as well. Okay, cloning vector as well. Clear? So, a good vector should have all of this. Normal ana yella vectors should have this ORI site. Okay, essential thing. If you are becoming a plasmid or if you are becoming a vector, idellame vanda konja simple ana oru topic. Okay, simple ana things like cloning site should be there. Cloning site na enna, where your restriction enzymes will be there, right? Cloning site where it can be single. It should be single, where your restriction enzymes can come and be. So, it is preferably single. Single sites it should be there, because if multiple sites are there, nama idu vanda vector vanda it will be cut into many pieces, right? If many cloning sites are there or this recognition sites are there, it is easy for all of them to go and cut. So adanala we will form many fragments. So adanala we should always think of having only single cloning site, most preferably single cloning site. So these are the characteristics of an ideal or a good vector. Now here we have to take this selectable marker as a complete, okay, complete topic by itself, students. So look here, in NCERT it is given cloning vectors. You know that plasmid and bacteriophages, okay, plasmid adukapram bacteriophages have the ability to replicate within the bacterial cells independent of a control of chromosomal DNA. Bacteriophages, because of their high number per cell and have a high copy number. So இதுக்கு வந்து spread ஆகுறதுக்கு chances ரொம்ப அதிகம், because அதுக்கு வந்து high copy number இருக்கு of their genome within the bacterial cell. So spreading becomes very easy. Okay, now some plasmid may have only 1 to 2. See, I told you, 1 to 2 per copy cell, whereas the others may have 15 to 100 copies per cell. 15 to 100 will be better for us. Okay, their number can go even higher. If we are able to link an alien piece of DNA with a bacteriophage or a plasmid, we can multiply its number equal to the copy number. Vectors used at present are engineered in such a way that they help in easy linking of the foreign DNA and selection of the recombinant DNA from the non-recombinant. Okay, understood? This is what is the 3 points that is required for a good vector. Okay, for a good student, what they should do? They should attend the class, they should come to the class every day, they should mark their attendance, they should talk to me, they should tell that, yes ma'am, I understood ma'am. That is a good student. Okay, good student. So, that is a good ideal vector. Okay, va? So, inga paaka students, the following are the features that are required to facilitate cloning into a vector. That means, if I have to clone into a vector, there should be certain sites that is required. Okay. So, first is origin of replication. This is a sequence from where replication starts. Molecular basis of inheritance la nama vanda detail DNA replication we understood. Adla we understood ORA site is required where the origin of replication is going to start. And any piece of DNA when linked to this sequence can be made to replicate within the host cell. This sequence is also responsible for controlling the copy number of the linked DNA. So, if one wants to recover many copies of the target DNA, it should be cloned into a vector whose origin ORI site supports the high copy numbers. If I want to make more, okay. Oh, Tamil thinkers edu Tamila. Okay, check pannanume. Okay, thank you Deepak, I'll check. Okay, now here selectable markers now students, in addition to the ORI, the vector requires a selectable marker which helps in identifying and eliminating non-transformant and selective permitting the growth of transformants. Transformation is a procedure through which a piece of DNA, students, transformation na enna, nama Griffith experiment ne transformation na enna, and the S strain vanda it went into the R strain and made the R strain virulent. Adha transformation. Oru piece of chinna piece of DNA vanda innoru cell kitta enter aachena adu vandu transformation. So in the selectable markers na it can do two things. It can help me understand if it is a recombinant or a non-recombinant. It can help me understand if it is a transformant or a non-transformant. Andha madri oru difference vandu clear I will be able to understand. Okay, transformation is a procedure through which a piece of DNA, okay, piece of DNA is introduced into a host bacterium. Okay, normally the genes encoding resistance to antibiotics such as ampicillin, chromaphenol, tetracycline, kanamycin, etc. are considered to be useful selectable markers. The normal E. coli cells do not have any resistance. So now we are going to learn last topic of the vector. Okay, cloning sites. In order to link the alien DNA, the vector needs to have a very few, preferably single recognition sites. Okay, for the commonly used restriction enzyme, presence of more than one restriction enzyme within the vector will generate several fragments which will complicate the gene cloning. The ligation of alien DNA is carried out by the restriction site present in one or two antibiotic resistance. So students, 2-3 conditions they have told that multiple recognition sites cannot be present, because multiple restriction enzymes also should not be present. Okay, let me Gopika is a fan page, okay? Gopi Sites is it's done by one of the senior students of yours, okay? I got to know very recently, okay? Senior student da, he messaged me on Telegram and then I got to know, okay? He is a very senior student. I think he's the last 2024 batch, okay? 2024 batch, okay? Okay, I will check this. I've taken screenshots, okay? Okay, students, so far understood this vectors, okay? Now we are going to learn about PBR 322. In the question one, the board, CBSE board favorite question, okay? CBSE board, CBSE board favorite question is the PBR 322. In the PBR 322, next for 10 minutes, I want your undivided attention, because in the 10 minutes, I am going to teach you something very, very important. How a vector can help us understand non-transformant, transformant, recombinant, non-recombinant. This is one of the complicated topics of this biotechnology. இதுக்கப்புறம் எல்லாமே ரொம்ப easy தான். Gel electrophoresis எல்லாமே easy தான். So அதனால I want next 10 to 15 minutes. Until I finish this topic, I want all of you to listen to me. Listen here. PBR 322 students, or vector ana, what are the things that I told you a vector should have? Ma'am, it should have an ORI site. It has an ORI site. Do you approve of it? Yes, ma'am, I approve of it. Okay. They told that it should not have multiple cloning sites. Ana, inga check பண்ணும்போது we have different restriction enzymes. SAL1 இருக்கு, BAMH1 இருக்கு, HIND2, CAL1, ECOR1, PVU1, PSTU1, PVU2. இது வந்து ரிப்பீட் ஆகாத ரெஸ்ட்ரிக்ஷன் சைட்ஸ். ஸோ அதனால எல்லா சைட்டுமே இண்டிவிஜுவலா இருக்கு. ஸோ தட் மீன்ஸ் cloning sites are less. Yes ma'am, approved. So second tick on the ideal vector. அதுக்கப்புறம் third thing they told was these people should have selectable markers. Markers na antibiotic resistant genes should be there, like your ampicillin, kanamycin, tetracycline. So adu madri PBR 322 has two restriction sites, right? Two selectable markers, ampicillin resistant, adukapram tetracycline resistant. So paakumbodhu எல்லாமே அல்டிமேட்டா இருக்கிற ஒரு ஐடியல் வெக்டர் தான். Correct or not? Okay? Correct or not? Do you agree that this is a good vector, or you will tell no ma'am it's a bad vector? I want to know. You will tell it's a bad vector or is it a good vector? Tell quickly in the chats. Because after this we are going to learn selection. We are going to learn selection. So I want all of your undivided attention here also. Okay? Good vector. Yes. So students, இப்போ இந்த selectable marker, if it is present, என்ன யூஸ் அதுதான் important. So அதனால இந்த PBR 322 use தான் we are going to understand. So first we will make three different hosts. Okay, host A, host B, host C. Okay va? See, now we are going to draw three sets of, okay, A, B, C. All of you can take one, you can choose A, B, C, whichever you want, you can choose. Okay, now A la vanda nama genetic material irukku, okay, B layum genetic material irukku, C layum genetic material irukku. Okay, B la vanda plasmid enter ஆச்சு. Okay, இங்க வந்து ஒரு plasmid gene of interest. Okay, this has happened. Okay, before that I want to tell you one thing. You just imagine we have PBR 322. Correct or not? Yes ma'am, we have PBR 322. So I am drawing it in this side. Okay, this is your PBR 322. This is the PBR 322. Okay, in the PBR 322 there are tetracycline and ampicillin resistance. Yes ma'am, correct. Okay, tetracycline and ampicillin resistant. Correct ma'am. இப்போ students imagine பண்ணிக்கோங்க, I want to add my gene of interest. This is a plasmid, right? Sorry, this is a vector. So this has to do the job of carrying the foreign DNA. So in the PBR 322 two regions are there. இது வந்து நம்ம amphicelline region. Okay, இது வந்து நம்ம amphicelline resistant region. Okay, இது வந்து நம்ம tetracycline resistant region. Okay வா? Imagine பண்ணிக்கோங்க students, ஒரு gene of interest இருக்கு. Okay, I have a gene of interest. Okay, I have a gene of interest. இது மாதிரி ஒரு GOI இருக்கு. ஆனா I inserted this GOI near the tetracycline resistance. What did I do? I inserted my gene of interest into the tetracycline resistance. Now you tell me if this tetracycline is going to be active or not. Quickly tell me in the chat box if this tetracycline is still going to be active or not. I inserted this gene of interest into the tetracycline region. Now you tell me if this tetracycline is going to be active or not. Yes or no? Yes or no? Quickly. Fast, 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 fast. Will students, this PBR 322, P for plasmid, B for Bolivar and R for Rodriguez, 322 is the sequence. Okay? No ma'am. I'm asking you that I inserted this in the gene of interest one. I inserted near the tetracycline resistant gene, where the tetracycline resistant gene was there. So now will the tetracycline resistant and the capability of the vector be active or not? Active or not? Will it be active? No ma'am, it won't be active. This is called as, see students, because I inserted something to one region, அது வந்து inactive ஆச்சு. So இந்த process க்கு we call insertional inactivation. What do we call? We inserted something that led to inactivation. So insertional inactivation. Insertional inactivation. Okay va? Understood this? Yes or no? Understood this? Yes or no? Insertional inactivation. Okay, that is, I took a gene of interest and the gene of interest வந்து correct tetracycline resistant gene ல வந்து insert பண்ணி அந்த tetracycline resistant capabilities, tetracycline resistant என்ன? Tetracycline medium இருந்தா it will be like I don't care bro, I am tetracycline resistant. அது மாதிரி தான். It will now I inserted something that led to inactivation. So that is called as insertional inactivation. Is this clear to all of you? If you understand this concept, this will be very easy for you. That is why I am explaining this concept to all of you. Okay, I want all of you to listen to me now. Now we are going to understand, very important, very important. Now we are going to understand எப்படி நம்ம vector வந்து it will be able to identify transformant and non-transformant, recombinant and non-recombinant. Okay, so start பண்ணலாமா? So இதுதான் நம்ம A. Okay, I want all of you to become one group. All of you tell me, are you A, are you B or are you C? Which side are you taking? Okay, whichever group you are taking, you have to answer for that question. Okay, you have to answer for that question, that is why. Whoever is taking A, raise your hands in the chat box. Ready? Okay. Now students, look here properly. Okay, look here properly. Okay, many of them are A, many of them are B, many of them, no one in C. Yeah, come on, someone choose C also. No, பாவம் அப்படியே ignore பண்ணிட்டா. A யும் B யும். C, I want some of you to take C. Come on, quickly. Okay, now look here. இது நம்ம cell A, B, C. இப்போ இதில் we are going to understand if they are recombinant, transformant or what. Okay, இது வந்து நம்ம cell A. Students, cell A ல has the plasmid entered inside? Has the vector entered inside or not? Yes or no questions. Very fast, I want all of you to answer. Come on, quickly. Let me see if I can. Tell me quickly. Yes or no? Yes. What did I ask all of you? What did I ask all of you? I asked you if a plasmid has entered or not. Okay, or any vector has entered or not. Your answer to that question is no, ma'am. Yedume enter agila, ma'am. So in the cell one, the students, it is non-transformant. In the cell one, it is actually non-transformant. Because nothing has got transformed into the cell. Transformation in one piece of genetic material has to enter in the cell. Non-transformant. If plasmid has not entered, then no, don't need to even think about recombinant. Okay. In the cell, students, in the cell one, what has happened? Has the plasmid entered inside or not? Yes or no question. Has the plasmid entered inside or not? Yes or no question. Come on, quick, 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 quick. It has entered inside? All of you think so? It has entered inside, ma'am. So, adanale idu vandu transformant aayirukkum. It will be transformant. Ana, it is non-recombinant. Why? Because there is no gene of interest. If there is no gene of interest, I have not made anything new. The vector just got inside. Now, can I say you are recombinant? He'll be like, where? Where am I recombinant? So, adanale, it has only become non-transformant. That means transformant. Transformation has happened. That means the vector has entered into the cell. A small piece of genetic material has entered into the cell. Ana adu vandu recombinant kediyadu. There is no gene of interest. So, adanale it is transformant and non-recombinant. In the cell paakumbodhu students, idu vandu it is transformant. Yes or no? Anyone disagrees? And it is a recombinant. It is transformant and it is recombinant. Correct, no? Yes, ma'am, I agree to this. Okay, whoever understands this, trust me, your entire topic, this biotech will be super duper easy. This is the only tricky part. Okay, idukapra nothing is tricky. So, students, now in the information use panni, I am going to separate transformant and recombinant because selectable markers job is to do that. So, now what I am going to do? I am going to plate. Okay, I am going to have an amphicoelin plate. And the amphicoelin plate, you tell me if A is going to grow or not. If I have amphicoelin resistance, I will aram se grow because I am resistant to it. If it's there, if it's not there, I don't have any thought about it. So, you tell me if I do a amphicoelin plating. Okay, if I do an amphicoelin plating, will A grow in that plate or not? Come on. And if I do a tetracycline, will A grow or not? Yes or no question. Will A grow in amphicoelin medium? Will A grow in amphicoelin medium? Think and answer. I don't want anyone to answer suddenly. Okay, making mistake. The answer is no, ma'am. Okay, no. Why? Because students, this doesn't have a vector only. It doesn't have your PBR322. It does not have any vector which has a selectable marker. So, on the resistant one, it does not have at all. Okay, it does not have that resistance only. If I am saying that, let's say something has entered into my body, I am able to beat 10 people, then I can say that yes, I have the resistance. Nothing is entered only, then how will resistance come? So, adanale A cannot grow, okay, A cannot grow in both the mediums. It is going to die in both the medium because these are antibiotics. It's going to kill the bacteria. So these are antibiotics, okay? So it cannot grow in both A and B. Try to understand. I'll teach you again. Try to understand. Then, now you tell me B, what will happen? The vector has entered inside. The vector has ampicillin resistance and tetracycline resistant genes. So will it be able to grow in ampicillin medium and tetracycline medium? Yes, it is okay to make mistake. Now only you can make mistake. You can't make mistake in exam. Okay. Tell me. Fast, fast. Okay, look here. It has ampicillin resistance, it has tetracycline resistance. So, adanale it can grow in both ampicillin and tetracycline medium. Right? It can grow because it has the resistant vector, it has the selectable marker, it has the resistance. So, adanale adu vandu it can grow in ampicillin medium and it can grow in tetracycline medium. Why? Because it has ampicillin resistant gene and tetracycline resistant gene. I am resistant to it, then why can't I grow in it? Right? I am resistant to, let's say, blue color. Okay, if I sleep in blue color the whole day also I will not turn blue color. That means what? I have the resistant gene, blue resistant gene I have. So if I have that gene, I can grow in that medium. I can grow in that medium. So adanala answer vandu S irukum. Now listen to me carefully. Listen to me carefully. I know many of you are making mistake, but see, here I have done insertional inactivation. What have I done? Insertional inactivation. So all of you told me before that, ma'am, since you did insertional inactivation, tetracycline gene will not be active. So I want you to think and answer this, okay? You have to only use the concept knowledge and understand this. So students, inga vandu insertional inactivation kapram and the tetracycline gene vandu damage aachu. Okay va? So now you tell me, will it grow in ampicillin medium? Think and answer, think and answer. If you answer only, then I'll have the confidence. Otherwise, I'll have to teach you multiple times. I want you to finish fast. Come on. Tell me this. Very good, very good. It will grow in ampicillin medium. Why? Because ampicillin resistant gene is still active. Ampicillin resistant gene is still active. Very good. Very good. Now you tell me, will it grow in tetracycline medium? Will it grow in tetracycline medium? Adukku chance irukka ilva? So, will it grow in tetracycline medium? Chance irukka? Illaya? Chance irukka illaya? Tetracycline medium வந்து damage ஆச்சு because of insertional inactivation. அது வந்து inactive ஆச்சு. So அந்த gene இருந்தா கூட they cannot still grow in tetracycline medium. No, why? Because it got damaged because of insertional inactivation. So அதனால they will not be able to grow in the tetracycline medium. Is this understood? Is this understood? Yes or no? This is the only tricky concept. That's why I kept telling you people, listen to every word that is coming out of my mouth. Okay, now you people got it. Now all of you are answering right. All of you are answering right. First you did not understand, now you understood. So basically I am able to, with the help of selectable marker na antibiotic resistant genes like your ampicillin resistant, tetracycline resistant, and the genes use panni eppidi transformant and non-transformant, recombinant and non-recombinant, adu madri oru classification how I can make it. So if the vector has entered into the system, I can say they are transformant. If they are not entered, then I can say non-transformant. So in the cell vandu non-transformant, in the cell vandu transformant, ana it is non-recombinant. Edukku ma'am? Because and the gene of interest vandu illa, vector la gene of interest illa. So adinala here it is transformant and recombinant, but ana inga vandu insertional inactivation has happened. Because of that, the tetracycline gene has got inactivated. More than this, students, clarity no one can give you. I guarantee you. Okay, people will copy from here, but no one can give you this clarity. So that's why I'm telling you, listen to me carefully. Clear? Recombinant means where the gene is there. Non-recombinant means where the gene is not there. Okay, Santosh. If you are making Jimmy and Sammy, and the Jimmy gene on the Sammy, that plasmid, it should be inside the plasmid. Illana, I cannot tell it recombinant because you did not make anything new. It is just the plasmid. Okay, just the vector. It's not new, it's not recombinant. Recombinant something new. Some cutting, chopping has to happen. So that is what is recombinant and non-recombinant. Transformant, something has transformed into the gene. That means a small piece of DNA has entered into another cell. So that is called as transformant. Clear to all of you? So this is actually your entire process. Okay. Praveen, sarcastic? Are you sarcastic? Okay, look here. In the PBR322 students, in the P stands for plasmid, B stands for Bolivar, R stands for Rodriguez, 322 stands for the sequence. Okay, va? These are the two people who found this, okay, sequenced. Okay, BPBR322. Okay, clear? Ha, white color, blue color, yes, yes, I'll come to that. Yes, yes, I'll come to that. Okay, va? Understood? Yes? Thank you, Deepa. Thank you so much. Okay, done. So, this is what we have understood. So, again I am not going back to this because again if I read NCERT then again you will have confusion. So, we will just stick to this entire slide vanda romba important da. So, make sure that you are revising this slide. Okay, va? Now, yeah, now I have to teach you about chromogenic substrate. Because students see in the process vanda romba hectic ana process, right? Tetracycline, ampicillin, continuous, okay? Continuous, I have to keep, doing the plating. அது வந்து ரொம்ப கஷ்டம். ஸோ அதனால நம்ம வந்து ஒரு சப்ஸ்ட்ரேட் யூஸ் பண்ணி பிகாஸ் ஆஃப் த கலர் டிஃப்ரென்ஸ் நம்ம எப்படி டிரான்ஸ்ஃபார்மன்ட் ரீகாம்பினன்ட்னு செப்பரேட் பண்ண முடியும்னு ஒரு ரிசர்ச் நடந்துச்சு. அந்த ரிசர்ச்சுக்கு அப்புறம் வி காட் டு நோ தட் நமக்கு வந்து ஒரு குரோமோஜெனிக் சப்ஸ்ட்ரேட் யூஸ் பண்ணி நம்ம எப்படி அந்த டிஃபரன்சியேஷன் பண்ண முடியும். இல்லைன்னா ஆம்பிசிலின் டெட்ராசைக்ளின் இது வந்து பிளேட்டிங் பண்ணணும், அதுக்கப்புறம் பாக்டீரியா வில் டை, ஆன்டிபயோட்டிக்ஸ், இட்ஸ் வெரி காம்ப்ளிகேட்டட். ஸோ தட் இஸ் வை தே கேம் அப் வித் another மெத்தட் which is called the chromogenic substrate. So students actually இதுல வந்து PUC18 or PUC8 we say, okay. In the plasmid that they are taking in reference, Anna, it is not mentioned in your NCERT. So I will also just tell you very like just the outer covering of it. Okay, just the outer covering of it. That is your PUC8. Okay, PUC8 or 18 also some people will teach you. PUC8. In the PUC8 there is one gene here which is your 𝐴𝐻 gene. Okay. In the 𝐴𝐻 gene, when the students, it will form what? The beta galactosidase, which again we have studied in your Lac-Operon concept. Okay, tosidase. Okay, beta galactosidase. Now, in this PUC18, so many cloning sites are there. Okay, EcoR1, BAMH1, your HIND3, romba irukku. Okay, Anna, what is going to happen? You will notice that in case this beta galactosidase, okay, in case insertional inactivation happens, that is in the 𝐴𝐻 region, okay, in the 𝐴𝐻 region, okay, let me just write it this way for you. Same like how I wrote tetracycline, I'll show you. In the 𝐴𝐻 region, students, or insertional inactivation has happened. Okay, that means one gene of interest has been inserted here. If it is inserted here, do you think it will still be able to form beta galactosidase? It will be still able to form beta galactosidase? No, ma'am, it will not be able to form. Correct? No? Okay, it will not be able to form. So, if insertional inactivation has happened, that means, example, foreign DNA has entered into the cell. Okay, imagine foreign DNA has entered into the cell. You use the same concept, students, and the concept ellame இங்கேயும் யூஸ் பண்ணணும். Okay, yes ma'am, can you please teach for class 10th? I'll try, dear. So look here, foreign DNA வந்து insert ஆச்சுனா, that means insertional inactivation has happened, then I can call them recombinant. Yes ma'am, you can call it recombinant. Correct? No, same thing like tetracycline only, just that we are changing the vector here. We are changing the vector here. So if a foreign DNA has entered, it will not allow the beta-galactosidase to form. With the help of the colors, I will be able to understand if the beta-galactosidase is formed, or if the gene is or the entire vector is recombinant or not. So in the foreign DNA, if insertional inactivation happen, then beta-galactosidase will not be formed. Okay, if it is not formed, remember, if it is not formed, it will give white colonies. Okay, white colonies. These are not dyes, they are substrate. Okay, chromogenic substrates. And in case this is formed, then it will give blue colonies. Okay, understood? So in a petri ஐயோ என்ன நடக்குது, இதுதான் நம்ம petri plate. So in the petri plate there will be some white colonies. Students, same tetracycline information you use and understand this also. Don't complicate in your head. If you complicate in your head, even if I'm teaching you detailedly also, you will still have doubts. Okay, understood this? Understood this? So if it is recombinant, that means insertional inactivation has happened, namakku vandu white colonies form agum. Okay, so this is my recombinant ones. Okay, here insertional inactivation has happened, that means gene of interest plus a vector has combined here. Okay, vector vandu form achu. Ana if I get blue colonies, okay, if I get blue colonies, that is an indication that it is non-recombinant. Okay, va? Non-recombinant. Clear? So this is with the help of chromogenic substrate. Clear? Understood? Clear, 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 clear? Tell me quickly in the chats. Okay, why is the attendance going down? Such important concepts, attendance will go down and then they will sit and cry. Okay, next we have to understand gel electrophoresis, which is super duper easy. Okay, super duper easy. So look here, next topic is gel electrophoresis, adikapuram competent DNA, and then we will be done with the chapter. Okay, are we done with the chapter? Let me just check through my notes as well. Yes, we have next gel electrophoresis, adikapuram PCR. Yes, adikapuram competent DNA. Yes, yes, yes. We'll be done in another half an hour. Okay. How will you get like that? Non-transformant, no, non-recombinant. Here no transformant. Non-recombinant, recombinant. That's it. Okay, user by. அதில் வந்து நம்ம transformant, non-transformant, இங்கே வந்து நம்ம மெயினாக ஃபோக்கஸ் பண்ணுறது recombinant and non-recombinant. எங்கெல்லாம் insertional inactivation நடந்திருக்கு, there it is the recombinant. Rest is all non-recombinant. Okay, here there is no transformant conversation. Okay, and even if let's say they are not, like if the vector has entered inside, beta-galactosidase will still be formed. It will form blue colonies. We want recombinant gene. We do not want non-recombinant ones. So even if 100 is formed, it doesn't matter for us. Okay. Yeah, okay, good. Okay, students, next வந்து நம்ம gel electrophoresis. ஏன்னா இந்த gel electrophoresis நம்ம already molecular basis of inheritance-ல detail we have studied. I told you before also, if I am sending you telling that you have to cut and chop these fragments, you're not doing the job and you're coming back, what will I do? I have to check it. So அதனால like undigested and undigested and digested strands வந்து separate பண்ணணும். Okay, so look here. Purified DNA is incubated with the restriction enzymes. Okay, guys, please focus here. If they have cleared need, good for them. Okay, don't fall for all this. Listen here. Okay, all those people come for attention. If a person has cleared NEET, they will be sitting in some medical college. Okay, good if he has cleared it, but you people don't have to go behind it asking what's your cutoff, which college, when did you, eat your food. You focus here. Okay, you focus here, please. Okay, he has cracked it because he has studied when he had to study. Now you people are not studying. Clear? Now look here. Purified DNA is incubated with the restriction enzyme. As a result, DNA வந்து digest ஆகுமே. Yes ma'am. What is this? DNA digest. These DNA fragments are separated by a technique called as gel electrophoresis. Gel electrophoresis. இந்த gel electrophoresis ல என்ன நடக்கும்? Gel electrophoresis ல students இந்த சைடுல வந்து நம்ம wells இருக்கும். என்ன இருக்கும்? Wells இருக்கும். இந்த சைடுல வந்து so negative, DNA is negative, so it will run against the positive charge. அந்த சைடுல positive charge இருக்கும். இந்த சைடுல wells இருக்கும். அந்த wells ல வந்து நம்ம sample add பண்ணணும். Okay, so here you can see agarose gel electrophoresis is employed to check the progression of the restriction enzyme digestion. That is, we want to understand if the digestion has happened or not. Buffering. One second, let me check. It's okay, no? It was just for a second, I guess. Now it's fine, right? Fine, right? Now okay, right? Yeah. One second, students. Have you all seen the video? Doctor salary after MBBS? When can you do this? Okay, so after MBBS you can do, which is around 3 years span. Okay, adukapra students kiruge they say adu one of medicine, adu one MD kaparam you can do. Yes, fine. Okay, fine, right? Okay, now look here. Agarose gel electrophoresis is employed to check the progression of an restriction enzyme digestion. DNA is negatively charged, so it moves towards the anode. DNA fragments are separated according to their size through the sieving effect. Okay, through the sieving effect of the agarose gel, a polymer extracted from seaweed. And the agarose gel when the polymer extracted from the seaweed. Okay. Now, the smaller size fragments move further and the process is repeated with the vector DNA. This is to check what, students? This is to check if the separation has happened or not. Okay, or the digestion we can say. Separation illana nama digestion nu solluva. So, digestion, okay, separation or digestion of the fragment. So, to know if they have actually digested restriction enzymes, have they actually digested the DNA fragments or not. Now understand, students, the smallest fragments, okay, and the smallest fragments it will move far. Okay, remember in school days and all, they used to tell, hey, you're chubby, how will you run? You're very thin, you can run. But Anna, that is all true, false, okay, because my friend was very chubby, she can run faster than me. So, there is nothing called as if you are little chubby, you cannot run. If you are very thin, you can run. Nothing like that. Air cannot take us and go, no. So, I used to be very, very thin. Very thin, okay. Very, very thin, students. Network issue, for me or for you? For me or for you? Is it okay now? Yo, that video went off, da. It's correct, no? It's correct only. So, look here. So, here the smallest will run farther and the largest, that is largest na enna students, idu vandu digest agala, the restriction enzyme has not cut it. So, it will be nearer to the well. Okay, it will be nearer to the well. Now, can I see it as such? No ma'am, you cannot see it as such. Okay, you have to dye it, right? You have to what? Dye it. So, what will I use? I'll use a carcinogenic, okay, carcinogenic dye, which is ethidium bromide. So, I'll use ethidium bromide, which is a bright orange color. Ethidium bromide, which is bright orange. I have to dye, then only I can see students. Illana, I cannot see the movement of the fragments. Okay, va? Yes, yes. Body shaming is a funny thing here. Okay. Yes. So, ethidium bromide, bright orange color. So, I will be able to see it under the UV rays. Okay, I will be able to see it under the UV rays. I will dye it with ethidium bromide, bright orange color. I will get it. Clear? Done, done, done. Okay. Yes. Okay. Good job. Okay. Okay. This process, students, I can call it elution. That is basically separation of the DNA fragments. Okay. Elution. So, this much you have to remember in gel electrophoresis. Clear? Now, next one is amplification of gene of interest. Inga vanda students, I want to tell you some more things in detail here, even though, okay, even though you don't have to know that much, but I'll tell you in detail for you to understand the whole concept. Now, isolation done, cutting done, separation done, amplification is left. So, now we have to do amplification of gene. Amplification of gene na enna? Thank you, Rohini. Amplification of gene, na, I have to increase the number, right? With the small, I cannot do. So, I have to increase the number. So, for amplification to happen, students, now on the PCR. Another PCR. PCR na enna? Polymerase chain reaction. Another polymerase chain reaction. I am using polymerase chain reaction. In the polymerase chain reaction, three steps are there. Okay, 3 steps are there, and the 3 steps enna, students, all of you remember we call bay. Bay we remember, okay, bay. All of you used to call in your friends and all. Hey, bay. Babe you call now also. Bay anna something before Christa. Theriyadu students, I don't know what is the full form of this. We used to tell bay. So, on the bay here, you remove here and add one D. So, it will become a day. In Tamil Nadu, everyone will tell, no, a day, enna day. Okay, adu madri tha day. So, day is here, D for denaturation, A for annealing, and E for extension. This is our PCR reaction. Now, students, in the PCR reaction, very pramadamana, okay, ஒரு பிரமாதமான ஒரு விஷயம் என்னன்னா இந்த டே வந்து temperature difference basisல நடக்கும். It's not like, let's imagine, okay, இது வந்து polymerase chain reaction. This is my let's say my PCR, my tube. Okay, now I add everything together to this. Now I cannot keep removing it, that after denaturation I'll remove it, after annealing I'll remove it, after extension I'll remove it. அது மாதிரி பண்ண கூடாது. So எல்லாமே செட் பண்ணி இந்த டியூப்ல ஆட் பண்ணினா the only thing I can do is temperature change. I can increase the temperature, decrease the temperature, increase the temperature, decrease the temperature. So just imagine if I have to make one person form some new strands, because I am doing amplification na enna number one increase pannum. So increase pandradhukku I need a polymerase. Now imagine I put DNA polymerase, it will become like one bhaji, it will become, okay, it will become like one okay, it will become like one enna students and the pappadam madri, it will become like one bhaji, it will get roasted. So adanala I will have to use someone who can tolerate the heat. Okay, because I will be keeping it 90 degrees Celsius. I cannot tell polymerase you go and do. Polymerase will become like one, okay, I don't know how to tell also, it will become, you know, it will get burnt. So adanala I have to use here Thermus aquatis. Okay, enna use panna a enzyme which is extracted from Thermus aquatis bacteria, which can withstand extreme heat condition. That is called as what? Taq polymerase. Whom am I using, students? Nama Taq polymerase. Taq polymerase. In the Taq polymerase, it can withstand any kind of temperature because I have extracted it from Thermus aquatis bacteria, which is a thermophile. Okay, it's a thermophile bacteria. That means it loves heat. Okay, it loves heat. Understood? Understood? So, in the denaturation la vanda almost nama vanda 90 to 92 degrees Celsius la idu vanda set pannina. annealing வந்து 72 degree Celsiusல. Okay, so இது தான் temperature difference. So you can understand, no, first I have increased the temperature, அதுக்கப்புறம் I am decreasing the temperature. Students shouting, my throat is gone. Okay, I am increasing the temperature, அதுக்கப்புறம் I am decreasing the temperature. Clear? Yes or no? Understood this much? Yeah. So students, in the temperatures when the vary agum, some places you will see 90 to 94, here you will see 55 to 72, extension you will see 64, adu madri varying temperatures you can see. In different books it will be given different temperatures, but ana oru general ana oru temperature. Okay, so that's why I am telling you, in the polymerase chain reaction first day வந்து எல்லாமே add பண்ணி only temperature regulation I can do. So I need to even add my polymerase in the starting of the experiment only. Students, denaturation என்ன, I have to make multiple copies. So அது copy பண்ணணும். To make the copies, I have to first separate my strands. So denaturation means where I am separating my strands. Okay, I am separating my strands. Annealing na, I have to add my primer. Remember in DNA replication also we had to add primer. If we don't add primer, this will not work. So I have to add forward primer and reverse primer. There what was happening? The primer was adding reverse and fragmentation was happening. Here fragmentation will not happen. Here it will be semi-conservative but continuous. Okay, so we will add forward primer and reverse primer. So idinale both the strands are going to be continuous. We are not going to get any lagging strand. Clear? So how is PCR different from DNA replication? Here we are going to get semi-conservative on a continuous strand. Semi-conservative on a continuous strand. Okay, va? Why? Because we are adding forward primer and reverse primer. அதுக்கப்புறம் extension la enna nadakum students? In the strands வந்து separate ஆனதுக்கப்புறம் we are going to get the new strands. Okay, understood? Like this I can make up to 1 billion copies. Clear? So this is the process. See here, denaturation, heat is provided, both the strands are separated. Adukapram I am adding reverse primer and forward primer where the annealing process will happen. Students, remember here we also call this as oligonucleotides. Okay, actually PCR vandu a detail on a lecture, like in masters and all we have studied this only as one topic. Okay, but Anna, if I teach you that much, that's all, you'll faint off. Okay, that's why I can't teach you that much. Okay, oligonucleotide primers. Okay, oligonucleotide primers all, see, remember nama tag polymerase ellame namakku synthesize panna mudiyum. So oligonucleotide primers will be added. We have reverse primer. Remember here, reverse primer. This is why we are getting continuous strands. See, this is why I told you molecular basis of inheritance you should know. Elena, you will not understand this chapter only. Reverse primer and forward primer, students. Then we have DNA polymerase where tag polymerase plus your deoxynucleotides. For energy you will need DNTPs. Okay, in the DNTPs used bunny we are going to form the new strands where extension will happen. So 30 cycles will take care. Remember, 2 to the power n. This is the number of cycles. So this is the formula to calculate your PCR. Okay, done? Understood? Can we proceed? Yes or no? It's almost going to be 3 hours. Very good. Excellent for students who stayed back. Okay, great motivation. Okay, the color is little different, no students, I don't know why. I think some brightness has been reduced. Okay, very good energy. Wherever Deepak goes, whichever channel he goes, wherever he goes, he has next level energy. Mangal Amma also. I don't know your real name, student, dear, but I have always seen your comment. You put all the jokes timing, everything. Very good, Karishma, Stancy, Janushika. Okay, very good energy. Okay, understood this much, right? Okay, so I don't want any of you to drop. Okay, and we should reach the target. Come on, we have one more topic and then we'll be done with this chapter. Okay, one more topic and we'll be done with this chapter. Okay, PCR is understood, all of you? Any student has any doubt? Next students, last topic that we are going to understand is competent DNA. That means, இப்போ வந்து இந்த vector வந்து it has to enter into the host. Do you think, okay, thank you, I'm improving in my Tamil, I have to improve a lot, but analum, I'm trying very level best to improve, okay. So students, here one thing you have to remember is, நம்ம vector வந்து host, okay, if it has to enter into the host வந்து accept பண்ணதுக்கு chance ரொம்ப கம்மி. Imagine randomly you are walking, you will enter into some neighbor's home, they'll hit you, no? So அது மாதிரி தான். But we have to make sure in the host வந்து நம்ம directly accept பண்ணதுக்கு chance, okay, அந்த வாய்ப்பு வந்து increase பண்ணணும். So அதனால தான் here we have to make the host competent. That means we have to allow the genetic material to enter into the host some way or the other. So here you see students, since DNA is hydrophilic, that means it does not like water, it is water loving, hydrophilic, okay, molecule, it cannot pass through the cell membrane because nama cell membrane la vandu lipid layer da, bilipid layer. So if it is hydrophilic, it loves water, but we have lipid layer, eppadi students enter panna mudiyum, so it cannot enter. So bacterial cells are made competent to make up the alien DNA or the plasmid as follows. That means to allow the entry of it. First one, the students, nama bacterial cell na treat pannam with using what? Divalent cations. Okay, using calcium, I will try to treat the bacterial cell at a specific concentration. Why? So the DNA can enter into the bacterium, because these will make small pores, okay. It will make small pores in the cell membrane. So like this, if the cell membrane is there, ayyo, like this, okay, this is the bilipid layer, okay, yeah, it is water loving. One second. Okay, one second. Okay, understood? Yeah. So இங்க வந்து என்ன நடக்கும் students? So here in the cation, this calcium ions la treat பண்ணினா automatically வந்து சின்ன pores இருக்கும், okay, they will make small pores allowing the entry of this genetic material, okay va? Okay, first thing what you will do, you will treat it with divalent calcium ions. So through the pores in the cell wall, they will incubate the cells with recombinant DNA technology. Adukapra நம்ம வந்து ice shock இல்ல heat shock ன்னு சொல்லுவாங்க. First you put it in ice, adukapram you will briefly put it at 42 degrees Celsius, that is called as the heat shock. So suddenly what will happen, you're basically confusing, okay, this person, telling what, okay, first you will put it in heat, adukapram suddenly you push it in ice, again you will put it in heat, adukapram you put it in ice. So what will happen, suddenly you will be like, what is happening, oh, it's cold, hot, it's cold, it's hot, it's cold. So on the confusion la this DNA will enter inside. Eppadi, okay, எது பண்ணாலும் plan பண்ணி தான் பண்ணணும், இது மாதிரி, okay, clear, understood this? So put them back on ice and bacteria takes up the recombinant DNA technology. There are other methods also students to introduce the alien DNA into the host. Enna adu? Okay, thank you so much Pujinal, okay, yeah, okay, these notes are made long ago dear. Other methods to introduce alien DNA into the host cell students, micro injections, okay, micro injections. நமக்கு வந்து இப்ப வேத் யூஸ் பண்ணலாம், so you can use வேத் to make notes, okay. All you have to do is, you should be a part of any batch or something. If you're a normal YouTube student, normally download the Vedantu app and get into Ved and ask Ved for notes. It will give you the notes, okay. It will give you flow chart, notes, just like how you use ChatGPT, Google Gemini, you can start using Ved, because Ved la வந்து நம்ம videos இருக்கும், and the practice questions இருக்கும், இந்த மாதிரி snips எல்லாமே add பண்ணி it will give you the notes, okay. So students, in this recombinant, okay, in this recombinant DNA is directly injected into the nucleus of the animal cell. So this is called as micro injections. Okay, micro injections are directly injection at the kichik. That's it. Okay, animal cell. Biolistics or gene gun. Okay, biolistics or gene gun, students. In this, the animal cells are bombarded with high velocity microparticles. Nama vanda chemistry la enna the students, Dalton's atomic theory, Thomson's what model? Thomson's watermelon, some model, pudding model, Thomson's pudding model. Okay. Thomson's pudding model, and the model ellame use panni, have you seen, and the bombarding they do, no, bombarding of gold particle, adu madri da idu. Okay, plum pudding model, yes. Okay, biologistic gene gun. In this, cells are bombarded with high velocity, high velocity microparticles of gold, tungsten, which are coated with the DNA. This method is suitable for plants. So question one, the students, inge a question varum, this is a MCQ. Remember, they will ask you which is the method that is for animal tissues and which is, sorry, animal cells, which is the one that is for plants. Okay, yes, clear? I will start biotechnology application after a break. Disarmed pathogen vector, students, they infect the cells and transfer the recombinant DNA into the host. Example, Agrobacterium tumefacium. I want all of you to remember this and keep TI plasmid. This is our application we are going to use. Okay, clear? Yes or no? Rutherford's experiment. Okay. How to balance board and neat, I have already made a video, dear. Please check it out. Okay, please check it out. So in the methods elama clear, students. So first we did heat shock, adukapram we did micro injections, adukapram we did gene gun, and then disarmed pathogen. Disarmed pathogen na nama people who are like very, okay, like mass people who will make problems, like rowdy baby types, that people we are sending inside, but we are keeping CCTV camera on their head. Now, will they rob? They will not rob. Will they do rowdyism? They will not do rowdyism, because police has kept CCTV camera on their head. Adu madrira. Disarmed pathogen na who can survive by themselves. Ana namma vanda and the characteristic vanda we have removed and we have told them you do now whatever you want. Adu madri. Okay. Basically, we are making the villain non-villain by taking his some of the benefits. Okay. So these are the methods that you have to remember. Clear to all of you? Nothing is there here, just competent host. Competent host la enalla irukum, students. First nama vanda, see, I am writing all these notes for you. Okay, I can chumma use NCERT and finish it off. Ana, I will not have satisfaction. I cannot sleep. Okay, you can go and check any of my class on YouTube. Every notes, every class I write. Okay, clear? So divalent cation, that is basically it will form pores, and through the pores the genetic material will be able to enter inside. Okay, GM will enter. Gopika Ma'am, army will enter. Okay, adukapram students, nama heat shock. Full confusion ana oru. This one, heat shock. First you keep it in heat. Okay, evlo degrees le 42 degrees Celsius le. Adukapram nama vanda ice la change panni. Adukapram pinne heat le. This idu madri oru cycle it will continue. Okay, adukapram students, micro injections. Micro injections. Micro injections na enna students? Micro injection na where we are directly injecting into the nucleus of the animal cell. Students, they will ask you animal cell and plant cell. Okay, so please make a note in your NCERT. It's a PYQ question. Okay, if you miss it, then gone. Okay, miss it, then gone. Clear? Very good. Okay, next one the students, nama biolistics illana nama gene gun. Gene gun la enna nadakum? Bombarding of the high velocity okay, gold and tungsten. Okay, high velocity gold and tungsten. Adukapram last we have disarmed pathogen. Students, here you can tell bacteriophage also, you can tell cancerous cells also, you can tell agrobacterium tumefacium also. Okay, clear? Clear? Yes, yes, very good money. All of you are present. Okay, good job. Raga, I'm not taking everyone's name, then it will become too long. Okay, so this is the method of your competent host. Okay, va? Ayyo, last topic on the students, bioreactor obtaining the foreign DNA. That means how are we going to large scale it? How are we going to make it big? How are we going to produce in larger scale? Okay, so here students see, these are the vessels in which the raw materials, okay, raw materials are biologically converted into specific products, enzymes, okay, using microbial, plant, animal or human cells. Bioreactors, na, it is big, okay, big vessels which are used to add in everything. Okay, temperature will be controlled, pH will be controlled, medium, nutrient medium will be there, everything. Why? Because I want to do a large scale production. I'm not thinking of tomorrow making 5 copies. If I'm making vaccinations, if I'm making let's say any kind of gene, if I'm modifying something, adu vandu large scale la nadakanam. And the large scale la nadakadukku I need big you know vessels where everything will be managed, everything will be mixing. Will you sit and mix from morning? No, you cannot do that. So adanale we have certain vessels which are called as bioreactor. One of state board's favorite question, bioreactors. Okay, so look here students, these are vessels in which the raw materials are biologically converted to specific products. So they are biologically converted to specific products. Okay, enzymes, etc. Bioreactors are used to produce large quantities of products. They can process 100 to 1000 liters of culture, okay, culture medium. A bioreactor provides optimal growth conditions like temperature, substrate, salt, vitamins, oxygen, ellame for the desired product. So அந்த மாதிரி ஒரு vessel imagine பண்ணிக்கோங்க, நம்ம வந்து adjust பண்ணணும். ஆனா இந்த vessel வந்து எல்லாமே regulate பண்றாங்க, but ஆனா a human intervention is required. A person will be sitting with a monitor to control, like he will be setting the temperature, everything and giving. Okay, so bioreactor will provide optimal conditions, okay, for the desired product growth. The most commonly used bioreactor is a stirring type, which is called as stirred tank bioreactor. What is it? Stirred tank bioreactor. So this is the one students, simple stirred tank bioreactor. It is usually cylindrical with a curved base to facilitate the mixing of the reactor content. The stirrer facilitates even mixing and oxygen availability. Alternative air can be bubbled through the reactor. The bioreactor ku edella irukku students, okay, thank you. Bioreactor ku inga motor irukku, foam breaker irukku, blade irukku, culture broth irukku, sterile air enter panna oru small pipe irukku, steam for sterilization, acid base for pH control, ellame irukku. Okay, iduna most commonly used one. Idu irukku, but ana idu vandu okay. Ana here you can see foldings are there where the gas, okay, gas entrainment will be there. Bioreactor has an agitator system, agitator na mixing system, oxygen delivery system to provide oxygen for the growth of the microbes. Adukapram students, foam control, because continuous mixing will lead to bubble formation, and the bubble formation vandu vacuole formation ku lead agum. So and the foam vandu nama break panniye aganum. Adukapram students, or temperature control system irukanam, pH control system irukanam, and sampling ports na periodically நம்ம வந்து அந்த culture withdraw பண்ணி check பண்ணணும், because we don't know what the machine is making, no, so we have to do this. Okay, you just have to understand the basic and the diagram you have to go through, because Neet la வந்து they can ask you diagram. More than that, they will not ask you match the following, what is present, what is absent and all. But then here you should just remember the they can ask you diagram based questions, like anything they can just hide and they can ask you what is this. Okay, they will ask you what is this. So you should know that it is sterile air. So last minute you can go through diagrams. I'll also teach you diagrams, so that time we can go through that, but otherwise don't have to break your head for this. Now students, next thing that we have to understand is, imagine, okay, I am like you know doing a project, okay, or let's take, imagine here we have students, no, Satish and Deepak. Okay, these two people are like, okay, we want to make a vaccination, we are going to do a next level large product, we are going to make 1000 liters of you know everything is already made, நம்ம வந்து ஒரு vaccine பண்ணக்க start idea வந்துச்சு. அதுக்கப்புறம் they have made it also, But ஆனா இந்த vaccine வந்து is it safe for the Indian habitat, is it safe for the people to consume, அதுக்கு ஒரு method இருக்கணுமே. அது method தான் downstreaming processing, where we are going to check if the product these people have made is actually can it be sold, can it be commercialized, is it safe for people, அதெல்லாமே நம்ம டெஸ்ட் பண்ணணும். First of all, Satish and Deepak made, so I would have already disapproved it. Banana, let's send it for downstreaming processing. Okay. So look here, it is a series of processes such as separation and purification of the product after the biosynthetic stage. The product is formulated with suitable preservative, because if it is commercialized, if it has to be kept, okay, be it drugs, be it medicines, be it cosmetics, idellame to keep it for a longer time நம்ம வந்து preservative add பண்ணோம். So அதனால suitable preservatives will be added. Such formulation will undergo thorough clinical trials. Okay, clinical trials will happen and strict quality control testing will happen. Is it safe for people to use it? Now students, certain like now in news I was seeing that, you know, certain tablets that people consume, in that tablets there are certain synthetic items which are not digestible for our body, and it will lead to the cancer formation. So adanale many of the medicines are going to be banned. So like that, many things are there. So for it we have to go through clinical trials. We will be using transgenic animals to do the clinical trials if it's a vaccine or an immunization. So like that, now we are going to understand in application chapter how we are going to use recombinant DNA technology. Clear? How we are going to use recombinant DNA technology. Clear? Okay. Students, questions are in the last, we will do it in the end. Okay, so okay, done. guys. Hello, hello, hello. Yes. Are we back? Oh, all of you were waiting. Such good kids I have got on Neet, yeah. Such good kids. Hello, Basingla. Yes. Edukada. Give me some time. Okay, I'll go and come. Huh? To drink one coffee also, you people won't give me time. Come, ma'am. Come, ma'am. Waiting. Good kids. Okay. Okay. Okay, ready? Next chapter on board. Okay, 1 minute. I'll just do your PPT. Energy regained. Yeah. Little energy, students. Little more energy is still to regain. Okay, but yeah, that much only energy is there. Yes. Start pannama. Okay, 1 minute. Yeah, just loading this. Okay. Will we achieve the target or it is too much target? Ma'am, target is too high, ma'am. Okay. Now, students, whatever we studied in biotechnology principles, அது வந்து எல்லாமே இங்க யூஸ் பண்ணு. Okay. Whatever you learnt there about recombinant DNA technology, gene cloning, எல்லாமே இங்க வந்து யூஸ் பண்ணணும். So, whatever, okay, 1 second. Okay, yeah, whatever we used, okay, whatever we studied, everything has to be utilized here. So, look here, biotechnology and its application, providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme, creating optimal conditions through engineering, downstream processing technology to purify. This is what we studied right now. Whatever we were giving them, we were giving it ultimate quality, purity. அதுக்கப்புறம் நம்ம வந்து அதை பியூரிஃபை பண்ணி டவுன்ஸ்ட்ரீம் பண்ணி ப்ராப்பராக ப்ரொசஸ் we were doing. Now in your biotechnology application, students, you have to study certain topics, okay, certain topics which are being taken from your strategies. All of you remember strategies chapter? Why can't I see your chats? Yes. So students, strategies chapter, yes, little tired money. Okay, strategies chapter la tissue culture, micropropagation, somatic hybridization, idellame they have been taken it from there and they have added to biotechnology application. So we are going to do NCERT line by line. Okay, as a traditional breeding technique failed to keep the pace with the demand to provide sufficient, fast and efficient systems to crop improvement, another technology which came into existence is called as tissue culture technology. In the tissue culture technology, na students, basically what does this tissue culture mean? It was learned by scientists during the 1950s that a whole plant could regenerate from the explant, that is any part of the plant taken out and grown, maybe a small portion of the plant, a small leaf, a small tissue, I can take it separately and I can grow it into a completely new plant. Okay, any part of the plant taken out and grown in a test tube under a sterile condition in special nutrient medium. This capacity to generate the whole plant from any cell or an explant is called as totipotency. That means a capacity of that portion of the plant to develop into a completely new individual. Now, if I cut the hand and keep the hand there, will it completely grow into a Gopika Ma'am again? No. And the potential வந்து நம்ம stem cells க்கு தான் இருக்கு. ஆனா it is not for every cell in our body. அது மாதிரி இந்த explant ஒரு small portion of the plant which can grow into a completely new individual or an organ. Clear? Now look here, you will learn how to accomplish this in higher classes, but here one very important PYQ question is here, students. It is important to stress here that the nutrients, okay, the nutrient medium must provide a carbon source such as sucrose and also inorganic salts, vitamins, amino acids and growth regulators like auxin and cytokinin. இது வந்து ரொம்ப important. Yes, your energy shouldn't go down, then my energy will go down. Okay, your energy shouldn't go down. Clear? So students, இது என்ன students? In the nutrient medium that I am providing, and the nutrient medium le I need to provide what and all? Carbon source, sucrose, okay, carbon source such as sucrose, inorganic salts, vitamins, amino acid, growth regulators like auxin and cytokinin எல்லாமே provide பண்ணிட்டு அதுக்கப்புறம் growth வந்து it is going to happen. Clear? So I am taking an explant. இதுதான் நம்ம explant, ஒரு small portion of the plant. We call it an explant. Okay. I am keeping it where, students? I am keeping it in a nutrient medium. இதுதான் நம்ம nutrient medium. So in the nutrient medium la இந்த explant வந்து grow ஆகும். ஆனா இந்த nutrient medium la என்னெல்லாம் இருக்கு? Carbon source இருக்கு. Okay, carbon source இருக்கு like sucrose. அதுக்கப்புறம் minerals are there. Vitamins are there. Okay, vitamins. Okay, vitamins are there. Then auxin and cytokinins will be there. Clear? Amino acids will be there. Everything to right place is not there. Okay, all this will be there in the growth medium, nutrient medium, so as to provide what? So as to provide the growth of this explant, and the potential or the capability of this explant to grow into a completely new individual is called as totipotency. Okay, yes, understood? Now, by application of this method, it is possible to achieve propagation of large numbers, okay, large number of plants in a very short duration. This method of producing thousands of plants through tissue culture is called as micropropagation. Okay, students, even I have done tissue culture in my masters. Adu vandu simple ana or lab illa. Okay, one test tube will be there, and the test tube la vandu nama nutrient medium we form. Now the formation of the nutrient medium, the most complicated process of tissue culture. So what we have to do, we have to measure every single thing, we have to measure, and not even 0.01% difference should not be there, because that will affect the nutrient medium. And we arrange it in like rows like this, and we keep it below the test tube. Okay, sorry, the tube light. Okay, because there is no natural sunlight, we will keep tube light, and we will keep it in a sterilized condition so as to allow its growth. And after few days, we will see slowly small plants emerging, and other one later we will separate it. Okay, so that is called a micropropagation. Each of these plants were genetically identical to the original plant from which they were grown. So we can call this what? Soma clones. Because students, they are all going to be identical, small pieces, no? So all are identical, so adinala we can call it soma clones. Many important food plants like tomato, banana, apple have been produced on a commercial scale using this method. Try a tissue culture laboratory, your teacher will make you understand. Okay, but unfortunately now we are not in that college, so we cannot go. So students, remember, tab is formed by tissue culture. Tab is formed by tissue culture. Tomato, apple, banana. Or you can remember bat. Okay, anything you want to remember, you can remember. Yes, few more topics to go. Okay, clear? Now look here. Another important application of the method is the recovery of a healthy plant from a diseased plant. Even if a plant is infected with a virus, the meristem, okay, meristem, that is the apical and the axillary of the virus. So students, basically nama enna oru unhealthy plant or a virus infected plant. Okay, for a virus infected plant, from that virus infected plant, you have to take the apical, you can take the apical meristem, and that apical meristem will not be affected. So using that apical meristem, I can form a healthy plant. Okay, I can form what? A healthy plant. Clear? Understood this? Yes or no? Healthy plant can be formed. Okay. Clear? Yes or no? Understood? Yes. So here, scientists have even isolated single cells from plants, and after digesting their cell wall, they have been able to isolate. So in the process, one I'll explain you separately. Okay, one second. Okay, we do not have slides, students, so we will have to borrow slides. Okay, you have to borrow slides. Look here. They are telling, first they told about micropropagation, that is understood, no, all of you? Micropropagation, explant, adellame okay dane. Should I write it again? No, large number of plants, yeah, large number of plants can be formed in short duration, genetically identified soma clones can be formed. Virus infected plant, from virus infected plant, just by taking the apical meristem, we can form a new plant, that also is understood. Now, next topic is somatic hybridization. Next topic is what? Somatic hybridization. Somatic hybridization. Somatic hybridization in a student, somatic hybridization. Basically, we are taking the protoplast. What are we doing? We are taking the protoplast. Okay, protoplast of the cell. So, example, one easy example I'll tell you. Students, they try to extract tomatoes, whatever characteristics of tomato was there and whatever characteristics of potato was there. Okay, they took it together and they made what? They made pomato. They made what? Pomato. But this pomato did not become successful. We make all this chapati curry, we make potato and tomato, but on the pomato one, they did not have all the essential characters that was required for the pomato. So, adinala idu vandu fail aachu. So, what is exactly happening here? Normally, this is the cell. Okay, inside the cell, you will have the cytoplasm and you will have the nucleus. So, I extracted the protoplast. Okay, I extracted the protoplast. Okay, extracted the protoplast. So, this is the protoplast 1. Protoplast 1 and this is protoplast 2. Okay, I extracted this two students, that is cytoplasm with nucleus. Adukapram, I am combining this. Okay, I am combining this. So, here I will combine this and I am going to get something called as a hybrid. Yes or no, will I get a hybrid? This is what I was teaching you so far in principles, that we are forming recombinant DNA technology. Okay, it was not tasty. Yeah, it was not tasty. I don't know pomato. I didn't try. Okay. So, here what is happening, students? We are taking the protoplast. Okay, we have to digest the cell wall. This will happen. Okay. Okay, this will happen, but adukapram protoplast eduthu we are going to form a hybrid. In the hybrid vanda we call it as what? Somatic hybrid. We call it as what? Somatic hybrid. So, this process will be called what? Somatic hybridization. We call it what? Somatic hybridization. Understood? Somatic hybridization. Clear? Yes or no? Digest the cell wall, then we take the protoplast. Okay? And the protoplast, we are going to combine it, we are going to get somatic hybrids. That's it. Very easy. We formed pomato, but unfortunately pomato failed. Okay? Pomato failed. Now, students, next topic is green revolution. Enna ma'am, in the green revolution. Green revolution succeeded in tripling the food. So, basically, to increase the food supply by 3x, green revolution was possible. In the green revolution was given by, okay, given by M.S. Swaminathan in India. Okay, Swaminathan. M.S. Swaminathan was the person who came up with green revolution, and worldwide it was given by Norman. Okay, this M.S. Swaminathan was the person who gave up this green revolution. In the green revolution, students, nama vande tripling the food. That means to increase, because see, everyday population is increasing. Okay, population is increasing, but we cannot the growth of or the food graph is not increasing. So, adanala we had to do green revolution, where we are using better pesticides, fertilizers to double the growth or triple the growth. So, to do 3x food supply, but yet it was not enough to feed the growing population. Even though students, we did tripling of the food, still it was not enough for the growing population. Clear? Now look here, increased yield have partly been due to the use of improved crop varieties, but mainly due to the use of better management practice. So, they are saying that how were we able to do 3x? How are you able to do 3x studying now? when you're sitting in the class because of the better management skills. If you would have studied this chapter, it would have taken you 5 hours to read NCERT, understand the concept, without any teacher's help, biotechnology principles itself. Anna, with better management, you are able to study 3x better. So here, better management practice, use of agrochemicals, okay, use of agrochemicals, that is fertilizers and pesticides. Adhikapram, however, for the farmers in the developing world, agrochemicals are often too expensive. Further increases in yield with the existing varieties are not possible using conventional breeding. So basically what is happening, students? Here what is happening? You should understand that to feed the growing population, number one, the green revolution we started. It was happening at 3x. But Anna, still it was not enough for the growing population. But ma'am, how did you make it 3x? How did we make it 3x? By using better agrochemicals. Okay, instead of using that normal, things, we could use agrochemicals. Second thing, students, by using high yielding varieties. Correct. Third thing, what? By better management practices. That I will do like this, if I do like this much crops can be formed. Better management practice. In the 3 methods used panni, nama the green revolution, tripling effect of food we have done. But Anna, it was not enough for the growing population. Clear to all of you? Yes or no? Okay, we can do fusion of protoplast, dear. Okay, you have studied events, this one, sexual events in fungi, have you studied? Fusion of protoplasm, fusion of nucleus, okay, that only. Clear? Yes or no? So, this is what happened. Okay, this is what happened. Now, somatic hybridization I taught you, pomato, green revolution also you understood. One second, green revolution, nothing students, only that 3x thing is there. Okay, GMO, GMC and transgenic, this is important. Plasmogamy, like that only fusion can happen. Fusion can happen like that only, clear? Okay. See students, plants, bacteria, fungi, animals whose genes have been altered. I told you, gene manipulation. That means, nama the gene alter pannumbodhu, we call that organism or the plant as genetically modified organism or genetically modified crops. Why? Because we manipulated the gene. If you don't manipulate the gene, then how will it happen? Clear? So look here, GM plants have been useful in many ways. Genetic modification it is, made crops more tolerant to abiotic stress. That means usually plants, when it is extremely cold, drought condition, very heat condition, the plants will wilt, the plants will die. But Anna, in the genetically modified plants, we have manipulated the gene in such a way, we have used recombinant DNA technology to make sure that this plant can survive any condition. Makes sense? Understood? So, reduced reliance on chemical pesticides. That means we have reduced the use of all the unnecessary chemicals on the plants, by which humans also die, plants also will die. And also students, when you use so much fertilizers, the soil quality also will reduce over time. Understood? Come on, I cannot see thumbs up in the chat box. Am I stuck? Okay, and we are very close to the target. Come on, let's achieve the target. Come on students, for my We Need Tamil Nadu students, this is a big target or what? One more person, come on. One more person and then we are good to go. Okay, I'm not stuck, no? I got, thumbs up should keep coming, otherwise I'll think I'm stuck. Okay, look here, made crops more tolerant to abiotic stress like cold, drought, salt, heat, reduced reliance on chemical pesticide, helped to reduce the post-harvest loss. Number one, the family, students, if you ask your grandparents or anyone like that, they'll tell you that, after the crops have been harvested also, lot of loss will happen. Many would have gone with weeds, many would have, during the process, many would have lost. So, post-harvest loss also we can reduce. And then students, increased efficiency of mineral usage by plants. That means what? We have to increase the efficiency of mineral usage. Plants will be like, okay, I have so much minerals, I will use everything. But Anna, by using a GMO plant, it knows the controlled effect that I want only this much, I will only take this much. So, the fertility of the soil also will be maintained. Okay, wow. This will prevent the early exhaustion of the fertility of the soil. Enhanced nutritional value of food. That is, we will increase the value of food. Example, when the golden rice. Next, we are going to study that. Okay. Next, we are going to study that. And students, this is a very important question for both. Okay. Okay. Both is a important topic. Okay. First topic. Okay. Whatever we studied so much, no. Whatever we studied so far, now we are going to use it. That is called as, first one is students, golden rice. So in the entire process, you might not understand looking at the image immediately, but first I will write, then we will do. So what was happening now students, many people were getting night blindness, okay. In one region, night blindness was becoming too much. So adanale they had to do golden rice. That means they understood that in the night blindness one, the main issue enna, it is because of the lack of vitamin A. And people were also getting fertility issues. So adanala they were like, okay, wait a minute. Ipo na vandu tablet kudthena, everyone, entire population people have to consume the tablet. Better than that is what? If I add it into a meal of theirs, that is, by adding that vitamin A into the rice, I can make a better efficient rice, right? That is what we have studied about recombinant DNA technology. If I can manipulate anything, then why can't I manipulate a gene? So, adu madrida. So, listen to me carefully. Here what was happening, students? Here, basically, your normal rice is there, no? To that rice, namma vanda, we are adding beta carotene. Ma'am, who is this beta carotene, ma'am? Students, this beta carotene is actually a precursor of vitamin A. Meaning enna? That means once it enters into our body, it will get converted to vitamin A. So it is a precursor of vitamin A. Now, from where did I extract this beta carotene? I extracted it from daffodil. This gene was extracted from daffodil plant. Can you see here? It is a daffodil plant. From there I took a gene. That gene was inserted into the plasmid. From the plasmid it got into the agrobacterium and then it is continued. So students, this gene I have got from the daffodil plant, which is beta carotene, which is a precursor of vitamin A. Okay, precursor of vitamin A. Clear? Why are you texting Ritika? Huh? Which PDF you want? Hridaya, which PDF you want? From that time you are asking one person, you have any PDF. Which PDF? This notes PDF you will get after the class. Okay, description box il irukku. Okay? Why are you torturing Pavam Ritika? Huh? Why are you asking Ritika? How will she know? Huh? Okay. So, here வந்து என்ன நடக்கும் students, நம்ம beta carotene gene வந்து we have extracted it from daffodil plant. Okay. In the daffodil plant, what we have extracted, அது வந்து we are going to add it to. Okay. We are going to, now we have to insert it. No, so we are going to insert it into a plasmid. Right. We are going to insert it into a plasmid. So, we will insert it into a plasmid. Okay. And then we will add it into the rice. Okay, so now rice plus beta carotene, rice plus beta carotene vande enna nadakum? Ma'am, you got the golden ticket, ma'am. So nama golden rice will be formed. Okay, in the golden rice, students, enna speciality? Vitamin A vanda, vitamin A will be rich. Students, so many chapters you are asking me to do. I will do everything one by one. You have to give me time. Okay. Vitamin A enriched. Next is cell unit of life. After that, I will do cell cycle and cell division. Vitamin A enriched. Now, whatever people were having like color blindness or any fertility issue, it got fixed because of this. Okay. Clear to all of you? This is easy. This is easy. So, this is the normal rice. This is the golden rice. Okay, this is the normal rice, this is the golden rice. Can we achieve the target quickly? Come on. Okay, clear to all of you? This is understood? Yes or no? Yes, yes, I'll do all the chapters. Okay, is this understood? Yes, ma'am, done. Years they might ask, dear, because only few years are there, no? So adanaala chances irukku. Chances adhigama irukku for them to ask the years. So adanaala just try to remember it in some way possible. Okay. Okay. Yes, done. Next one, the students, நம்ம Bacillus thuringiensis, okay, BT cotton. This was the first one that was introduced in India. Okay, so pest resistant plant. That means if if some cotton or anything like that, like a brinjal, idellame, if it is getting affected by a pest, நம்ம வந்து gene addition use பண்ணி எப்படி we can tackle it. Okay, so pest resistant plant, how we can form. So that is what we are going to understand with the recombinant DNA technology only. Now students, see, understand here, pest resistant na enna? Normally or insect vanda continuous it is attacking a plant. Okay, this insect is continuously attacking a plant. Okay, like the cotton was attacked by your bollworm. Okay, all that. So same way, normally students, like this your cotton will be. Okay, have you seen cotton? How many of you have seen cotton? They'll be like, ma'am, we have seen cotton candy. That I know. You tell me cotton. இதுதான் நம்ம காட்டன், கரெக்ட் தானே? இதுதான் நம்ம நார்மல் காட்டன். ஆனா வில்லன் என்ட்ரி ஆச்சு ஸ்டூடெண்ட்ஸ். வில்லன் வந்தேனா இந்த காட்டனை, this is the cotton like bud. Okay, this is the cotton bud. இந்த cotton bud-ல வந்து it will start making like this. Okay, it will start entering like this and it will make some pores. Okay, how many of you have seen cotton? Thank you, MR. Okay, yes. So students, insect is continuously affecting the plant. Now I know recombinant DNA technology. Adu vandu use pannanume, like your knowledge you have to use. Okay, you have to use the knowledge in your NEET examination. I knew you people will tell cotton candy. I did not expect even one student to tell, yes ma'am, I have seen cotton. Okay, some of them are the exceptional students there of my class. Okay, I know you will tell, ma'am, I have seen cotton candy. Okay, whatever you have seen, no. So look here, what is happening, students? There's a bacteria. So how do we do this? Okay, how do I protect this plant? So we got to know that there is a bacteria, okay, which is called as Bacillus thuringiensis. Bacillus thuringiensis. Okay, in the Bacillus thuringiensis, students, for its self defense, edhukaga self defense kaga it will protect. Okay, it will self defense kaga it will form some crystallized protein which can attack. Okay, which can attack and do certain things. Understood this much? Yes or no? For its defense, it will form some crystallized, okay, proteins, which is inactive stage, which is in an inactive form, but it can get activated in alkaline pH. Okay, it is inactive, so it will not harm the bacteria. So as a mode of self defense, it was releasing a crystalline protein which has the ability to actually attack the insect. So adu vandu namakku use pannalame. Yes ma'am, you can use it. How am I going to use it? I am going to use this crystalline protein which is your BT toxin. Okay, cry protein also we can say. In the cry protein use panni, I am going to protect this cotton from all the dipterians, leptoterians, coleopterans, ella insectume I am trying to protect it. Okay, so students, here try and understand. This is how your normal cotton is. Once the insect infects us, this is how cotton will happen. So it is a very big loss, right? It's a very, very big loss for a farmer. You imagine yourself as a farmer, you will not be able to tolerate this. Yes or no? You will not be able to tolerate this. So we have to use this crystalline protein somehow to help. Okay. Now, what is going to exactly happen? நமக்கு வந்து beetles இருக்கு, flies இருக்கு, butterflies இருக்கு, worms are there, எல்லாமே it's a threat to the cotton. So அதனால students, in the BT toxin வந்து, how are the farmers using it? Okay, we have extracted this BT toxin from the bacteria and it will be like a powdered capsule form. அது வந்து தண்ணியில mix பண்ணிட்டு இப்படி sprinkle பண்ணணும். Okay, we have to just sprinkle like this. So once we sprinkle it, students, it will go and fall on the cotton. Yes, ma'am, it will go and fall on the cotton. On the cotton, there will be larvae. On the cotton, who will be there? Larvae will be there. Okay, on the cotton, who will be there? Larvae will be there. Okay, larvae will be there. So this larvae will consume it. Yes, ma'am, very smart, ma'am, you have become. Larvae will consume it. இப்போ இந்த larvae consume பண்ணும்போது students, இந்த BT toxin வந்து it is in inactive stage தான். Okay, it is in inactive. Just because you mixed it with water, it will not become active. So it will now enter into the larvae. It has entered the gut. Entered the gut. They say mid gut. Entered the gut of the larvae. இப்போ larvae gutல students, the twist of the story. இந்த larvae gutல வந்து alkaline pH இருக்கும். என்ன இருக்கும்? Alkaline pH இருக்கும். So because of that alkaline pH, என்ன நடக்கும்? இந்த inactive BT toxin வந்து suddenly it will become active. Okay. So in the BT toxin வந்து it has become active. Okay, wait. BT toxin has become active. BT toxin has become active. Okay, wow. It has become active. So now what is going to happen? Okay, now what is going to happen, students? It is going to get into the larvae's gut. Okay, imagine பண்ணிக்கோங்க students, இதுதான் நம்ம larvae gut. Okay, very fancy ana gut I am drawing. Okay, okay, இதுதான் நம்ம larvae gut. இந்த larvae gutல it will go and do perforation. Perforation na என்ன? Pore formation. Perforation na என்ன? Pore formation. So அது வந்து it will form pore formation, and because of the pore formation, it will lead to lysis. Okay, so whatever is outside will start entering inside, which will lead to the swelling of the cells, and it will lead to lysis. Okay, swelling of cells, and it will lead to lysis of cell, ultimately leading to the death of the larvae. Understood this? Easy? Easy? Easy peasy lemon squeezy. Tell me quickly. Okay, wow. It will do perforation. Yes, I'm in 11th, ma'am. I'm confused to take biomaths or pure science for NEET. Don't take biomaths, yeah, it's difficult. It's difficult. Understood this? Understood, understood? Yes, good job, good job. Good job, good job. Okay. Yes. Done, 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 done. Okay. Done, done, done. Okay. Okay, okay, okay. Bhavana, you joined very late, Bhavana. Class is almost going to get over. Okay. Okay, done. So, pest resistant plant, this is what is going to happen to the larvae. Now, students, remember lepidopterans. So, here you can see it forms insecticidal, toxic insecticidal protein. Why does this toxin not kill the bacillus? And the question normally it should come, no. Why, ma'am, this toxin is not killing the bacteria itself? Why is it not killing Bacillus thuringiensis itself? Because அது வந்து inactive formula இருக்கு. In the presence of alkaline pH மட்டும்தான் அது வந்து active ஆகும். So அதனால, so it is toxic insecticidal protein. Why does this toxin not kill the bacillus? Understood. Actually, the BT toxin protein exists in inactive protoxin, but once the insect ingests it, okay, it will become active. Now look here, which are the insects that is going to attack it? Lepidopterans, okay, that is basically your tobacco budworm, armyworm. Coleopterans like your beetles, okay, coleopterans, adukapram students nama dipterans, okay, dipterans which are going to be a mosquito. Bacillus thuringiensis form protein crystals during a particular phase of their growth, okay. Now here you can see surface of the midgut epithelial cells la poi adu vandu perforation, it will form pores and that will lead to entry of other substance from outside leading to lysis, okay, leading to lysis and death of the organism. Understood this? Any doubt here? Any doubt here? Here you have to remember PYQ point of view. PYQ point of view, some things are important. Okay. Alkaline pH. Alkaline pH na nama basic pH. Okay. So specific Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into several plants like cotton, brinjal, etc. Why does it not harm us also? It does not harm us because even we do not have alkaline pH. We have acidic pH. Adanala, if we consume brinjal or lady's finger which has been, sprayed with Bt toxin, we will also not get affected. Clear? Now look here students, the toxin is coded by a gene, okay, and the toxin one, it is coded by a gene which is named as the CRY, okay, CRY. And here you have to remember PYQ point of view, rumba important. Many years PYQ you can go and check, this question is there, okay. What is it? CRY1AC and CRY2AB controls the cotton bollworm, okay, that is CRY1AB controls the cone borer, okay. So someone is very bored, so only one person will be there, that is CRY1AB, okay. AB will be very bored, okay, like that you remember, cotton borer, okay, corn borer, CRY1AB. CRY1AC and CRY2AB will be bollworm. Clear? Thank you. Okay, ma'am, help me take. Okay, Karthik, we will talk about it later, okay, it will distract the class, so we will talk about it later, okay, your decision we will do about it, okay. This is normal cotton versus the, the insect attacked cotton. Is this understood? Anyone has any doubts? Please ask me right now. Please ask me right now. Later you will ask me in the comment section, then it will be difficult for me, okay. Okay, next we are going to a very, very important topic of this chapter, students, gene silencing, okay. Gene silencing, are you ready? Okay, this is very important. Most of the students don't understand it at all, so adanala I'll go slow here. So students, next topic is called as RNA silencing. Illana RNA like suppression nu sollala, and the gene vandu mute pananum. இப்போ எந்த ஜீனை மியூட் பண்ண students in our body also, in eukaryotes also, you know that we have something called as gene regulation. நம்ம cellular defense-க்காக நம்ம வந்து certain genes will make it inactive at certain point of time. In molecular basis of inheritance, we have studied gene regulation, but we have studied for prokaryotes. But you know, in eukaryotes also this process will happen, and நம்ம gene regulation வந்து 4 steps illa it can get regulated. Here, that eukaryotic cellular defense we have used it to protect tobacco plant. Idhukku ma'am tobacco plant, because students in the tobacco plant main oru issue enna oru nematode 𝐂𝐴LDO incognita, and the nematode it was infecting and it was causing root knot disease. And the root knot disease protect பண்றதுக்கு fertilizer, pesticide, weedicide எல்லாமே யூஸ் பண்ணிட்டு it's not able to be protected. So அதனால we use something called as RNA silencing. Without allowing it to form translation or go into translation, not forming protein will help it to not express. That is the main goal. Okay, listen here. So what we were doing, students, here we are talking about a nematode which is your 𝐵 incognita. So this is your nematode infested plant. Okay, so normal root it will be like this. Okay, this is our normal root. Okay, okay. இப்போ அது nematode infested root ஆனா like this, root knot disease, because of which students the plant will not get enough transport of any nutrients or anything. Okay, so because of the plant will get affected. And tobacco is a very earning, like it's a very major earning point. So autofill, if you want to leave, you can leave, dear, you can leave. Okay, you don't have to spam. Students will come to class, they don't understand how much teachers will get disturbed. Yes, definitely. Okay, so students, look here, nematode. So what we are doing, this is called as your root knot disease. Okay, in a root knot disease. Because of this root knot disease, nama tobacco plant it was completely getting damaged, and it was because of a nematode. Enna students, and the nematode, what is the name of the nematode? Nematode was Melidogyne incognita. The pronunciation is a little different from everywhere. So it is Melidogyne. Okay, so this was the one. Now remember, this nematode, what it was doing, it was attacking the root, and because of that, what happened, students? It was causing root knot diseases. In the root knot diseases, because of which tobacco was getting affected. Now we have to protect this. Yes, ma'am, you have to protect it. How am I going to protect it? Students, the process of protecting this is called as RNA silencing, or I call it what? RNA interference. Okay, we are allowing the interference of an RNA, so we can also call it RNA silencing. This is an important topic here, so make sure that you're listening. Okay, RNA silencing or RNAi. Clear? இப்போ இது என்ன நடக்குது students? It is actually a naturally occurring mechanism in eukaryotes. Naturally occurring mechanism in eukaryotes. Okay, it's a naturally occurring mechanism in eukaryotes as a part of our cellular defense. Okay, as a part of our cellular defense. Anna, how do we utilize that is important? Yes or no? Yes, ma'am. So all eukaryotic organisms, students, all eukaryotic organisms use this as a cellular defense. All the organisms use it as a cellular defense. This much is clear. Now we are going to get into the little complicated part. Okay. Now what I am going to do? I am going to do silencing. Silencing of a specific mRNA. Now how do I do the silencing of the specific mRNA? I have to make sure that this mRNA stops and it does not go for translation. Because if it goes for translation, then adukapram protein formation kandipa nadakkum. So adinale and the translation process la vanda nama discontinue pannam. Eppadi? With the help of double stranded. This is the question la varum, students. Double stranded RNA. With the help of what? Double stranded RNA. Double stranded RNA. Now, double-stranded RNA is going to be the one. So, in the double-stranded RNA, students, one is going to be the sense. One is going to be the sense. Okay, one is going to be the antisense. And this antisense is the one that is go and attach and help us in the stopping the translation. Okay, one strand will be sense strand, one will be antisense. Understood this much? Now, how do I take this double strand, students? Two sources are there. One source is by a viral genome. Okay, infection with a viral genome. So, I get the RNA. One more with mobile genetic materials. That means the moving genetic materials. Okay. Mobile genetic material which are called as transposons. Yes, very good energy. Awesome. Awesome. Karishma, Mangamal. How do I pronounce your name, dear? Okay. Are we going to reach the target? Close to the target? Yes or no? Okay. So basically, students, this is what is happening. RNA or RNA silencing, it's a natural process that happens in eukaryotes, which is actually a part of the cellular defense. Now, why are we using this process? Namma tobacco plant la root knot disease is caused because of a nematode. And the nematode DNA it should not express. If it forms mRNA also, we should stop the mRNA. We should do silencing of the mRNA so it does not do translation. And the translation process kappuram, okay, because protein will be formed, protein will express. So adanale we have to stop the translation. But Anna, we cannot switch off and switch on any process just like that. So adanale we are going to use a double stranded RNA where the antisense is going to attach to the mRNA and make sure that it does not work. Why? Because we know that complementary strands will not work, it will not go into translation. So that is what we are going to do here. Okay, so here okay, 1 second students. Okay, I'll duplicate this only. Okay, and we will just draw one box which will make you understand the whole process. Okay, actually this process, students, it has lot of things. Okay, it has like agronut proteins, siRNAs, very complicated process it is very high level process also. So adanale, okay, Kamali, okay, adanale we cannot understand the whole, like you cannot understand it at one. If you go in detail, there's lot. Okay, so what is happening, students? Here we have nematode specific gene. Okay, number double stranded RNA the hero material. Okay, it is the hero material. So remember, in the double stranded RNA one is going to be number sense strand. Okay, adukapra innonu vandu anti sense strand. In the anti sense strand it is going to go and attach because it will be complementary. Okay, okay, this is one. Okay, now what is going to happen, students? Suddenly the nematode will be there. No, villain nematode will insert its DNA. It will put its DNA inside. In the DNA vanda it will convert to mRNA. Ana and the time nama double stranded. Okay, nama in the double stranded RNA, one of the strands will come and attach to this fellow. Okay, so now can it do translation? No ma'am, he cannot do translation, ma'am. Okay, it cannot do translation, ma'am. Practice, demon, practice. How can you keep all this content in the brain? Practice. Okay, so in the double stranded RNA will silence this mRNA, and that is the process. Okay, silencing. So in the double stranded RNA, students, it will attach and it will make sure that no translation happens. So translation illana protein formation nadakadu, so obviously it will not express itself, so the root knot disease also will not happen. Clear? Okay, va? Understood this? Yes. Okay, I'm going to sit for a minute. Just look and let me know. Oh my God. Yes, tell me. Done? Yes, ma'am. Done. Pakka, ma'am. Understood, Pakka? It's a little complicated topic. That's why I'm asking you multiple times, or else I would have moved on past it. Okay? Okay, good. Good job. Okay, done. Students, remember, if you want to learn this concept in detail, okay, and the details there will be 2-3 words that will be used, like risk, RICK, okay, then dicer, okay, dicer is the one to cut this, okay, like this multiple words will be there. So don't be like, oh my God, where is all this word coming? RNA induced slicing and all that, okay, signaling and all that. So risk assembling will be there, dicer will be there. Remember that these words are all there, but it is not required for you because it's not in your NEET syllabus, nor you are going to continue to learn this syllabus as well. Okay, so adinala we are not going to tell, but in case you watch a video or something and you notice it, please remember that it is all there, just that we are not filtering it out and teaching. Okay, now pest resistant plants, several nematode parasites, a wide variety of plants and animals, a nematode called Melanogyne incognita, MELOD. Did I put I or here? Wait. Should write correct spelling always. Nematode வந்து okay. Okay, now here going back again. Okay, so look here students, here incognitia, okay, infects the roots of the tobacco plant and it causes RNA interference. Okay, so RNA interference takes place in all eukaryotic organism as a method of cellular defense. So I'm not going to stress again on NCERT. We have already studied everything of NCERT, but only important words, okay, which involves silencing of a specific mRNA due to a complementary double stranded RNA. இதுதான் ரொம்ப முக்கியம். Okay, இதுதான் நம்ம PYQ point of view ல முக்கியம். Okay, wherever I've put PYQ, make sure you study it. Okay, tomorrow the notes will be available for you. Okay, now molecule that binds to and prevents translation of the mRNA. So basically என்ன பண்றாங்க, preventing the translation of the mRNA. Okay, the source of this complementary RNA could be from an infection by virus having a RNA genome or a mobile genetic material. So here the source, how I am getting this double stranded RNA, is from these two materials. Using agrobacterium vector, nematode specific genes were introduced into the host plant. The introduction of the DNA was such that it produced both sense and antisense RNA. In the host cell, these two RNAs being complementary to each other, they formed a double-stranded dsRNA that initiated RNAi and thus silenced the mRNA. Clear? That's it. That's it. Done, done, done. Understood? Is there anyone who hasn't understood? Okay. I don't know, Hema. Because of my students. Okay. Yeah. Okay, now students, medicine. Okay, medicine. Remember, in around therapeutic medicines, around 30 of them is found, but only 12 is sold in India. But the first therapeutic medicine that you have to study is about your one and only your insulin. Okay, insulin. So this is your islets of Langerhans. Okay, recently only, I hope all of you have seen my chemical coordination video, islets of Langerhans. Okay, in the highlights of Langerhans one, the students, நம்ம pancreasல இருக்கிற endocrine activity. Okay, it does the endocrine activity in the pancreas because pancreas வந்து ஒரு heterocrine gland. It has exocrine and endocrine activity. So on the endocrine la islets of Langerhans வந்து it forms your insulin. It forms what? It forms insulin. So what does insulin do? It is basically hypoglycemic hormone. That means it will reduce the glucose level. Okay, so it will நம்ம glucose level வந்து it will reduce. Okay, but ஆனா ஒரு problem என்னன்னா நம்ம பாடியில, okay, நம்ம பாடியில imagine there is no let's say there is no enough insulin. Okay, our body is not able to form enough insulin. Then what is going to happen, students? We'll have to take injections from outside. Okay, we'll have to take injections from outside. Yes or no? So students, here the main problem is if the insulin is less and the glucose is not getting converted, then we will go to a condition called as diabetes mellitus. Diabetes mellitus. Okay, in the diabetes mellitus condition enna nadakum students, main issue enna, here the deficiency will happen of the insulin leading to the person having more glucose in their body. So adukku vandu we have to take outside injections. So what they were telling, they were like, okay fine, we can form synthetic insulin, and they started forming insulin, but they started extracting it from, they started extracting insulin from cattle's, okay, cattle's and pigs. And the problem was, students, everyone was not okay with it. Okay, everyone were not okay with it. Why? Because the main issue was this was causing allergic reactions. Because it was extracted from pigs and cattle, it was causing allergic reaction. It was causing allergic reaction in people. So adanale they were like, okay fine. But you can ask me, no ma'am, why didn't you make insulin pills, ma'am? Okay, why don't you make insulin pills? But the problem is, students, insulin vanda, if I make insulin tablets, The issue is insulin is the protein hormone or a peptide. It's a peptide. Peptide hormone, na, it will go into the body. It will go into the stomach. The stomach has peptide breaking, pepsinogens and everything, which will break the peptide, no, proteases and all of that. So it will get broken down. So adanala I cannot use the peptide hormone. Yes or no? Correct or not? Agree? Agree? Ha. Allergy, not irritation, allergy. It caused allergy. Clear? So far good? So it's a peptide hormone, that means it's a protein hormone. So obviously it will be consumed by the proteases in the stomach. So it cannot be taken orally. Cannot. Cannot be taken orally. Clear? So we have to take it intravenously. Injection only we have to do. Now ma'am, what is the solution ma'am? You told so many problems. Yes. The solution is genetically modified insulin. Now students, insulin structure வந்து நமக்கு important தான். So நமக்கு ஒரு A chain present ஆயிருக்கும். அதுக்கப்புறம் நமக்கு ஒரு B chain present ஆயிருக்கும். And in the A chain and B chain will be connected with the disulfide bonds. Okay. எப்படி connect ஆயிருக்கும்? With the help of disulfide bond. And this A chain and B chain again it will be connected with the help of a C chain. With the help of what? With a C chain. So அதனால இதுதான் நம்ம insulin structure. So நம்ம synthesize பண்ணும்போது இந்த structure வந்து we have to keep in mind. So many people tried many things and it did not work. Then came a company called as Eli Lilly company, which is now also there. Okay, Eli Lilly company. In the Eli Lilly company வந்து they were the first American company that told that wait, we have to find a way to make humulin or synthesize. Okay, we have to synthesize this insulin. Ana the main barrier that they were facing is students to synthesize or to assemble. Main problem enna to assemble the C chain. The main issue that they were facing was to assemble the C chain. Issue that they were facing was to assemble the C chain. Then they realized, okay, that okay, wait a minute, we don't have to assemble the C chain because in the diagram or in the structure it is only present during the inactive stage or the protoxic stage. Protoxic stage, okay? Okay? Yeah. So during the active stage in the C chain it is not doing any function. So adanale what we can do is we just need the A chain and we need the B chain with the help of disulfide bonds. Problem sorted. Anna eppidi ma'am, how are they going to form this? Right. So assembling the C chain was the issue. That was removed because the C chain is only present in the proactive stage. Proactive stage. That means during the active stage, we only have these two people. Okay. Now, how do we form the students? With the help of E. coli. Now, we are taking E. coli, 2 E. coli and we are going to make the A chain and B chain. Okay. Again, recombinant DNA technology. Okay. So, this is one E. coli. Adukapra, here one more E. coli. Are you all okay? Yes. Energy is there. I know it's been long, but Anna, we have to do it, right? Students in quota are sitting for 13 hours with their books. So, adanala, even we need to at least sit for 5 hours to show that we are not weak. Okay. Students, A chain will be smaller than B chain. Okay, remember this. Because it will have less amino acids. Okay, ultimately we will form this. Okay, we will form this. Clear? Understood? Okay, they will be joined by disulfide bond. So this led to the formation of a very, very important thing, students, which is called the humulin. Okay, which led to the formation of a humulin, which had a A chain and a B chain. Any doubt? Any doubt here? Okay, understood? Yes, yes, yes. Okay, so active stage mein only this A chain and B chain is there, students. Okay, by the end of the class, all the languages are coming in my mouth. Okay, Hindi, Kannada, what not. Okay, A and B chain is the only thing that is there in the active stage. Inactive stage, the entire chain is there. Okay, yes. One more important topic is gene therapy. Are we ready? Yes, gene therapy. Come on, just stretch yourself. Okay, just stretch yourself and then we are good to go. We are good to go. Yes, okay. Breaker again. Why da again breaker? We just now finished a break now. Yeah, last second topic. We have still more diagnosis and transgenic animals. Okay, ready. So students, gene therapy is basically where we can, let's say, taking the default gene, we can add the right gene, and that is what is gene therapy. Okay, and the madri oru method that gene therapy. Okay, so the first successful case of gene therapy, first successful case of gene therapy happened in 1990. Okay, which was mainly the gene therapy had happened for a disease which is called as SCID, severe combined immunodeficiency. Okay, deficiency disorder. So, severe combined immunodeficiency. In the severe combined immunodeficiency, students, happens when a person is deficient with, okay, which gene is deficient in them, or enzyme, we can say deficiency of ADA, adenosine deaminase. In the adenosine deaminase one, the students, it helps with immunity. Okay, yes, 4 years old kid. Okay, very good. Okay, it was in a 4 years old kid. Okay, students, this ADA or adenosine deaminase deficiency one, it leads to mainly what? It leads to immune problem, that is because it helps in the maturation, differentiation of the lymphocyte. So it plays an important role, plays an important role in the immune system. Okay, plays an important role in immune system, also in lymphocytes maturation and other things. So, adanala idu vanda absent aayirundena students, then obviously the person will have a immunodeficiency. Now, how do I fix it? So, doctors, see, many methods were there. First one vanda enzyme replacement. Enzyme replacement therapy. Enzyme replacement therapy. This is the first method. Okay. Ana in the ella method mein students enna aachu, idu ellame oru solution da, but ana oru permanent solution kedaiyadhu. These are all temporary solution. So enzyme replacement therapy, adukapram bone marrow transplantation. Bone marrow transplantation. Ana see, everything has an expiry date. Okay, it will not continue. It has to be continuously been. Bone marrow also at one point it will get over, right? So adanala bone marrow transplantation ellame it has a certain time period, okay, time period. So adanal these are all temporary solutions. So enzyme replacement therapy happened bone marrow. Ana last one was gene therapy. But students, gene therapy also, if it is like, this is the best method among all of this. Ana, if let's say gene therapy should be a solution or a permanent solution, na, then we have to do the gene therapy, that is we adding or exchanging the defective gene at a very embryonic stage. If you need a permanent solution, it has to be done at a embryonic stage. Okay, va? At a embryonic stage. Clear? Done, done? Yes or no? Permanent solution can be gene therapy at an embryonic stage. Okay. Understood? Understood? That is the solution. How will I answer, Karthik? It's up to you, bro. Up to you. If I tell something and you do something and then you don't get some, why? Why all this problem? You surely select. Okay, done? Yes. Okay, this is done. Here students, see here. Presence of pathogen. Nama early method, if normal, let's say, what to tell, if I want to, let's say I want to do any test, okay, I'll first give blood test. Basic traditional methods ellame blood test, urine test, serum test. Ana, let's say I want to know if a person has cancer, AIDS, I can do ELISA technique, I can do PCR. So that is your molecular diagnosis. How much we have improved in the field of biotechnology, how much we have upgraded ourselves in biotechnology is what you're going to understand. So look here. Presence of a pathogen is normally suspected only when the pathogen has produced a disease symptoms. Yes or no? You will only know you have a fever until your symptom starts coming out. Otherwise, you will never know. So, by this time, the concentration of the pathogen is already very high in the body. However, at a very low concentration, let's say I have a bacteria which is at a very lower concentration. How do I know if it is in my body or not? Okay. So, or virus. Okay. Or virus at a time when the symptoms of the disease are not yet visible and can be detected by the amplification of their nucleic acid by PCR. So, PCR use panni I can amplify, so I will get to know if even if I have a low bacteria virus count in my body also, with the help of amplification I will get to know, I will be able to increase the amount for me to understand about this. Okay. Clear? What are you people talking? What are you people talking? Huh? Have you all marked your attendance? cDNA, complementary DNA. Can you explain how PCR can detect very low amounts? We already know PCR, how they can detect, students, by doing the amplification. It is being used to detect mutations, okay, detect mutations in genes in a suspected cancer patients too. It is powerful technique to identify many other genetic disorders, again like HIV, okay, or your AIDS. So, PCR is a technique that we can use as well. Okay, next one, students, a single stranded DNA or RNA tagged with a radioactive molecule is allowed to hybridize. So, this is called as autoradiography. This topic I have taught you detail I have taught you, okay, in your molecular basis of inheritance. So, those who haven't watched that lecture, please go and watch. In MBI, we have understood that we can do autoradiography. So, basically, students, understand, nama ELISA technique, PCR techniques, ellame, it will help us to understand, because nama old, okay, in the before, we will realize that we had only blood, serum, urine test. Ana ipo vanda we have ELISA, PCR technique, autoradiography. Adu madri high tech we have findings to understand if there is any problem or not, right? Yes or no? So, you should understand that DNA, Abhinaya and Demon, can you not sit and talk here? This is not WhatsApp. Okay, a single stranded DNA or RNA tagged with radioactive molecule is allowed to hybridize. Students, we all know, no, VNTR probe. VNTR probe. I hope all of you have seen molecular basis of inheritance, so I'm just going to read it out. The clone having the mutated gene will hence not appear on the photographic film. This also I have told you that normally நம்ம வந்து let's say AGAATTC. Okay. Now I have done a complementary gene against this, that is my VNTR probe. Okay. A வந்து T, G வந்து C, T, T, A, A, G. ஆனா என்ன ஆச்சு ஸ்டூடெண்ட்ஸ், இது வந்து mutated ஆச்சு. Okay. This has got mutated. Okay. Here. Okay. This has got mutated into G. Now what has happened? Wait, I'll write it with another color. So we know that it is mutated gene. So here what will happen? I send this piece of probe. In the piece of probe, it will not go and attach here, and in the frame, okay, in the autoradiography frame, I will be able to spot this, that here a mutated gene is present. Understood? This is the modern techniques. Okay, modern techniques. So ELISA is based on the principle of antigen. Again, I'll teach you ELISA in human health and diseases. Antibody antigen interaction. Infection by pathogens can be detected by the presence of antigen proteins like your glycoproteins, etc., or by detecting the antibodies synthesized by the pathogen. So antibiotic, antibody. Okay, hurry, you will get it in how many days over? 10 days is over. 10 to 15 days it will take, because it has to be delivered at your home, no? Okay, depends on the distance also. How far is it? Like, where is your place and everything. Okay, last topic, students, transgenic animals. Okay, last topic is transgenic animals. All of you ready? Yes. Okay, come on. Some of you have not marked your attendance, which is very upsetting. Okay, we are standing for so long and teaching, you need to mark your attendance. Okay. Okay, we'll start. Last topic, transgenic animals. Okay. What ethical issue, dear? Biopiracy, bioprocessing, that is there. Don't worry, that is there. Ethical issues are there. Okay. Yes. Okay. Animals that have had their DNA manipulated. So I told you this, no? The DNA which has been manipulated, those animals are your transgenic animals. Transgenic rats, rabbits, pigs, sheep, cows and fishes have been produced. Why are they, students? So why are we using transgenic animals? Namakku vanda vaccination test பண்ணதுக்கு இல்லனா immunization test பண்ணதுக்கு, toxicity of any products to test, we have to use it on transgenic animals. I cannot use it on you people, no? So adu madri tha. So look here, why are these animals being produced? But here there is a PYQ question that has to be highlighted. Although over 95% of the existing transgenic animals are mice. Okay, so that da nama 95% organisms la transgenic animals la highest use vanda rat da. Okay, normal physiology and development. Transgenic animals are used specifically designed to allow the study of how genes are regulated, how they affect the normal functioning of the body and its development. Example, study of complex factors involved in growth, such as insulin-like growth factors. Ella growth factors mein nama padikum bodhu we cannot put injection on us. So adanal we have to inject it on transgenic animals, which is cruel, but ana there's no other way. Okay, by introducing genes from other species that alter the formation of this factor and studying the biological effects. Okay, biological effects that results information is obtained from the biological role of the factor in the body, how the body is changing, எதெல்லா change ஆகுது, is it safe, is it not safe, அது எல்லாமே. So normal physiology. அதுக்கப்புறம் students, study of diseases. Many transgenic animals are designed to increase our understanding on how genes contribute to the development of the disease. So to study a disease, we have to use a transgenic animal, right? That is to understand, okay? To understand, these are specially made to serve as a model for human disease, so that the investigation of new treatments, so this is done by students, so we can investigate new treatments for the disease is made possible. Today, transgenic models exist for many human diseases such as which and all, cancer kaga, everyday research is happening for cancer, students. Anna, we are not able to find that one way to, deal with it. Ria? Yes, deal with it. So, cancer, cystic fibrosis, rheumatoid arthritis, Alzheimer's, all this we have the models for it, okay? So we have the models for it. Adukapram students, to test any biological products. So in the paragraph, the most important paragraph where your PYQs are going to come. Clear? So understand here, medicines required to treat certain human diseases can contain biological products, but such products are often expensive to make. So transgenic animals that produce useful biological products can be created by introduction of a portion. That means by manipulating the gene, we can make the transgenic animal form that biological product, okay? And the biological product formation vanda and the transgenic animals use panni form panla instead of synthesizing or making it in the lab. So which codes for a particular product such as like a human protein, okay? Very important, human protein alpha-1 antitrypsin. Students, this question is going to come as human hormone, human protein, human enzyme. So remember, it's a protein, okay? It's a protein, clear? Used to treat emphysema. Treat what? A chronic disorder caused by continuous smoking, okay? Smoking disorder, clear? Emphysema. Similar attempts have been made to treat phenylketonuria, so treatment to treat PKU as well, and cystic fibrosis. But in 1997, okay, important, in 1997, transgenic cow called Rosie produced human protein enriched milk. What was it? What are human protein enriched milk, which is around 2.4 grams per liter. This is also important, PYQ. Okay, they'll ask you the quantity, how much was that? 2.4 grams in per liter, human protein enriched milk. Because human protein, how are you going to get in milk? So that was, made by Rosie. The milk contained the human alpha lactalbumin and was nutritionally a more balanced product for human babies than a natural cow milk. Clear to all of you? It is a human protein, not a fat, okay? Not fat or anything else. So it's a human protein. Clear? This is very, very important. I've used different colors to make you understand. Next one, the vaccine safety, which is one of the most common use of transgenic animals. Vaccine safety, we all know, right? Transgenic mice have been developed or used for testing the safety of vaccines before they are used on humans. Transgenic mice are being used to test the safety of polio vaccine, and this is not from now, students, it is from years, okay? If successful and found to be reliable, they will be replace the use of monkeys, okay? Now it is not used much, but analum, lot of monkeys are tortured, okay? Now, ma'am, are you teaching NCERT? Yes, of course. What else am I teaching? Okay. Chemical safety testing, this is known as toxicity, okay? To test the toxicity or safety testing. This procedure is same as that of the testing toxicity of drugs. Students, before, nama shampoos le, nama perfume le, it used to be written animal tested. Ana ippa vanda they don't write that. And they write that it is not animal tested. Why? Because people have become more, people have started understanding how difficult it is to be a transgenic animal. So, adanale, they have told that you are not supposed to test it on animals because the cruelty on animals is too high. Nama transgenic animals, if you see the videos or documentaries of how these animals are treated, it's very inhuman. So, adanale, people do not consume anything where it is written animal tested. Ana vaccines, drugs, medicines vandu nama control la irukadu. So, adanala adu vandu tested agum. The procedure is same as that is used for testing toxicity of the drugs. Transgenic animals are made that carry genes which make them more sensitive, okay, which will make them more sensitive to toxic substance. And students, nama transgenic animals use panni nama ear pinna, அது வந்து create பண்ணலாம். So அது மாதிரி நம்ம genes modify பண்ணி we can do parts of our bodies as well. Okay, like ears and all of that. Then non-transgenic animals, they are then exposed to toxic substance and the effects are studied. So இங்க வந்து we are increasing the toxicity, their sensitivity, sorry. We are increasing the sensitivity towards toxicity. Okay. What are we doing? We are increasing the sensitivity, okay, so they can react to toxicity more, okay, or toxic substance more. So this is what we are doing in this process. Is this clear to all of you? Last topic for the day, okay. Last topic for the day. Ready? Yes or no? Ready, ma'am? Not ready, ma'am? When if you're not ready, I have to do it. Okay, I'll do it. Yes. மீண்டும். Yes, come on. Ready, steady, go. Let's continue. Yo, no, student, sorry. 1 second. Okay. Ready? Yes. 1 second. Okay. Now students, imagine, okay, I made basmati rice. You imagine. Okay, I made basmati rice. ஆனா ஒரு foreigner, okay, ஒரு foreigner Kevin வந்து அந்த basmati rice the patent na I have the power over that product. I can sell it, I can commercialize it, I can put my brand name, எல்லாமே பண்ண முடியும். Okay, இப்போ வேதாந்து வந்து ஒரு brand. அது வந்து it is owned by these people like CEOs and co-founders. அது வந்து no one else can use that name. No one else can make another brand with Vedantu name. அந்த மாதிரி ஒரு patent. We need to get the patent. So there is many a times what happens in plants in agriculture வந்து நமக்கு வந்து dwarf varieties. Okay, we know Mendel, right? We can form wrinkled, yellow, green, எல்லா கலர்ஸுமே ஃபார்ம் பண்ண முடியும். நம்ம வந்து cross பண்ணணும், test cross பண்ணலாம், back cross பண்ணலாம். So அது மாதிரி lot of methods we have. Okay, monohybrid, dihybrid cross. So the variety that I have already made of basmati rice, adu use panni I can make dwarf varieties, I can make tall varieties. So people will tell that, hey, this variety I only made, I need patent for it, because if you get patent, then you can ask money, right? You can ask money, you can ask big MNCs telling that you buy my rice, you give me this much of crores. Okay, so in 1 second. Okay, actually this one is not, the light is flickering. 1 minute. Okay, coming in 5 minutes. I will just ask that security uncle to fix it. Yes, students, I'm back. Okay, yeah. Okay, so last topic, students, biopiracy. I told you, no, patent. So what has happened, see here, basmati rice through the US patent and the trademark office, this allowed the company to sell a new variety. Okay, did I get cut here? Oh, first part is not there. Here only main part is there, don't worry. So here you can see, students, nama new variety, the company was able to sell a new variety of basmati in US and abroad. Okay, and abroad. This new variety is not actually a new variety. The new variety of basmati was actually being derived from a Indian farmer's variety. That is, Indian basmati rice வந்து நம்ம cross பண்ணிட்டு, dwarf cross பண்ணிட்டு, like with a dwarf plant, semi-dwarf variety. So, what did we do? It was crossed with a semi-dwarf variety and claimed as an intervention to the novelty. The patent extends to functional equivalence, implying that other people selling basmati rice could be restricted by the patent. Several attempts have also been made to patent uses, products and processes based on Indian traditional herbal medicine. Students, nama normala paakumbodhu ipo lot of companies vanda, oh, turmeric soap, turmeric with neem soap, turmeric with eucalyptus, turmeric with saffron. Ana idellame nama ayurvedathile idu vanda already prevalent ah irunduchu. So idu vanda new invention kedaiyadhu. Ana idla main enna, இது எல்லாமே abroadல USல வந்து very casually it has been sold because this is all Indian Ayurvedic. அங்க Indians வந்து they have to find out, okay, இது மாதிரி ஒரு இது இருக்கு. But this is all sold in every other places telling that this is very Ayurvedic. Like example, turmeric and neem. Okay. If we are not vigilant, we do not immediately counter this. Exactly. இப்போ packing வந்து கொஞ்சம் டிஃப்ரெண்டாக இருக்கலாம். ஸோ அந்த பேக்கிங் பார்க்காம நமக்கு வந்து turmeric, neemனு we will not even understand. It will be written in different way. It will be written in different format. So அதனால we will not understand. So like that, lot of patent cases happen every single year. Okay. Look here. Biopiracy is the term used to refer to the use of bioresources. Okay. To the use of bioresources by multinational companies. Multinational companies and other organizations without, okay, other organizations without proper authorization, okay, from the countries and the people concerned with it, okay, concerned with it, be it anything you are using, okay, anything you are using, everything vanda, okay, everything vanda, it is important for us to ask permission, take permission. Even many multinational companies also use a lot of things which has already been found. Okay, so adanale adu vandu konjam என்ன சொல்ற machine formatல technology use பண்ணி கொஞ்சம் modifications பண்ணியா they cannot get patent for it because it's already been found. Okay, most of the industrialized nations are rich financially but poor in biodiversity and traditional knowledge. In contrast, the developing and the underdeveloped world is rich in biodiversity and traditional knowledge. Traditional knowledge related to bioresources can be exploited due to the development of modern application. Now students, you all know how much AI, right, your ChatGPT and your Gemini that we were all loving at one point of time, and now also many of us love it. You realize how much of AI created shorts are there on YouTube and Instagram. You will not even understand it is AI. Usually I feel like if it is created by AI, or symbol at least irukanam for people to understand if it is real or false, because I see hundreds of cat videos, baby videos of AI created. ஆனா அது கொஞ்சமே AI video னு தெரியாம I like the video. அதுக்கப்புறம் I see the comments, it's an AI video, because அந்த actions, அந்த dog actions, eye movement எல்லாமே ditto to ditto humanly இருக்கும். So it is very dangerous. It's a world that is going to be more dangerous, where people are going to misuse AI. Right? People are going to take our faces, people are going to take all our information, and we are going to all you know, it's very, very scary. So, adinalada AI created videos, be it pro version, whatever version it should have a logo. It should have a watermark so people are aware that it is an AI video. And the madri filtration, if it is not brought about in our country in another 2-3 years, then exploitation is going to be extreme. Okay, exploitation of the content is going to be extreme, and it is very dangerous for all the women, kids and men out there who are working, who are in high professional levels, how much they create this AI video. Okay, so here students, it can save time, effort and expenditure during the commercialization. There have been growing realization of injustice, inadequate compensation and benefit sharing develop because of this biopiracy. So, idla vandu only biopiracy is important. There's nothing else that you have to break your head for. Okay, okay, answer this question students quickly. Okay, again I'm telling you, Saturday, okay, 7 p.m. there is going to be a special session which is about all the NCERT scientists, because you know in 2025 one unexpected twist came about all NCERT scientists, and teachers also were shocked. So adanale we are going to do all NCERT scientists. I don't want any of you to miss. Make sure to share it with your friends. I want to see maximum attendance now itself. After this class gets over, go and mark your attendance there. Okay, then only I get the motivation to come to class. Okay, yes. Now look here students, this is the BT cotton varieties that was developed by introduction of toxic gene of Bacillus thuringiensis is resistant to plant nematode, insect predators, insect pests, fungal diseases. 2020 question. Good, good. Yes ma'am, I will attend. Very good. Thank you, Mohammed. Yes, think and answer, don't answer wrong, then I'll have to scold you people. Okay, then I'll have to scold you people. Okay, I'm going to check the attendance. Let me see. Oh, come on students, we can do more by liking the video. Okay, smash the like button, and then in the comment section later you can let me know that you were there or not. Okay, you can just put Deepak present, okay, Karishma present, Adamadri. You can also put your attendance in the comment section with your name and how the class was. Okay, insect pest, very good. Next question students. It's too bright, no? 1 second, let me do something. Let me. Okay, this is better, no? This will come properly. Match the following column and set the correct option. BT cotton, adenosine deaminase deficiency, RNA interference, PCR. Okay, PCR. I'm hungry students. Did you all have your dinner? Okay, I'm too hungry. C. Okay, BT cotton vanda Bacillus thuringiensis. Okay, only one option is there. Very good. So you can do it with one, but Anna, we will do it with all. Okay, adenosine deaminase deficiency, gene therapy, RNA interference, cellular mechanism, PCR one. Okay, very good, very good. Okay, after the session, okay, option C, correct. Okay, adukapra next question. RNAi is a mechanism to silence genes with the help of so nice. You people are all waiting to have dinner. Done, na? Yes. Easy question. Answer fast. Nah. Hey, double stranded RNA, double stranded DNA, how it will happen? I told you sense and antisense. Okay. Double stranded RNA. If I have to silence mRNA, if I send DNA, how will it happen? It will be double stranded RNA. Yes or no? All of you are copy pasting your friend's answers. Your friend also doesn't know. You also don't know. Two monkeys are sitting in one class. Okay. Avloda. See here. See here, double-stranded RNA. Double-stranded RNA is the whole process. Okay, see here, prevents the virus, all eukaryotic cellular defense, and it is done by your transposons, prevents the translation of mRNA. What are we using here? Sense and antisense RNA, that is basically your double-stranded RNA. Double-stranded DNA, say what you will do? Huh? Yeah, only one person told correct. Everyone else is wrong. I'll do more questions of principles in the next class, okay? Now it's too late. So, golden rice is genetically modified crop plant where the incorporated gene is meant for biosynthesis of. This question is not there. This has been removed actually. This you answer. The first human hormone produced by recombinant DNA technology is. First, yes, today's mission is accomplished. Correct, correct. Okay, correct. Understood? Yes, very good. Insulin is the answer. Okay, insulin is the answer. Done, done. Okay, yes. The first clinical gene therapy was given for treating chickenpox, rheumatoid arthritis, adenosine deaminase, diabetes mellitus. Diabetes mellitus. Thank you, Demon. I want to see all of your comments in the comment section with your attendance, okay, if you were present or not. I want to see which are the genuine NEET aspirants who are there, okay. All of you come for 1 hour and then you will be like, yes ma'am, I'm a very serious NEET aspirant. It will not work out, okay. Quota mein there are students sitting for 12 hours, 13 hours and it is important, okay. Yes, the answer is going to be adenosine deaminase deficiency, clear, which causes SCID, okay, which causes SCID. Last question, use of bioresources by multinational companies and organizations without authorization from concerned country and its people is called as what? Same NCERT line they have just copy pasted here. Okay, 2018 question, same NCERT lines. Same NCERT line. Yes, answer. Yes, biopiracy. Very good, very good. Okay, so what I want in your comment section, I want your name, okay, present ma'am, and how the class was. Okay, yes. And students, now also NEET test series, you will get it for an affordable price. I know last time some of you did not get. So, adinale, I told you I will do something else as well. So, I am planning that. Let's see what will happen. Okay, another few days I will get an idea on what to be done. Okay, yes, great. Okay, I can see a lot of students joining now. I think 10 o'clock, many students are just using phone and they're just watching all the lives. Okay, so thank you so much, Pasangla. See you all in the next video. Till then, take care. Bye-bye. Make sure to subscribe, like the video. Every like, every subscribe that you give is a very, very big reward for us. You know how we started from 0. 3 months back we were at 0 and today I have okay GMAs. I have 12,000 GMAs in my channel. So adanale I want to grow it bigger. I know only you can do it. So adanale every time you go somewhere, every time you meet someone, make sure to share the channel. Okay, if you bring 4 to 5 people to the channel, that itself is a big contribution for us. So make sure that you're a part of this channel and the part of this entire beautiful community of very active students and mentor and everything. And yes, physics class is going to happen on Friday. Okay, mostly on Friday. Ray optics will be completed and sir will also do one-shot recorded videos as well. So your portions get completed. Okay, by December I'll give you a planner, what to be done, how much will be completed in the channel, what all are the other things that we are going to do. Okay, so Pasangla, so much planning is happening for all of you. All that I need is your energy to attend every day. Be present. Okay, be present. Give me that energy. Show me that power that you had from the starting. Okay. So thank you so much. Make sure to let me know in the comments how many of you were present and see you all in the next video. Till then, take care. Bye-bye.